Please come on to the dais and have a seat, please.
For you to see that.
Dr. Veda Rao, our principal and dean, Ramaya Medical College. Dr. Madam, vice principal, Ramaya Medical College. Our registrar, Dr. Pratap, Ramaya Medical College. And our registrar, Hemant Sir, Ramaya Medical College. Thank you, sir. And our in charge, CRL, Dr. Vijaya Mysore. May I request everybody to be standing, remain standing for our Ramaya anthem. Everyone to be seated. Now I request Dr. Vijaya Madam to please take the
Good morning, everybody. We are very happy to be conducting this Karyotyping and FISH workshop in collaboration with the Association of Medical Biochemists of India. Hearty welcome to all our dignitaries. We welcome Dr. Veda Rao, our principal and dean. We welcome Dr. Shalini Nui, our vice principal. We welcome Dr. Pratap, our registrar academics. Welcome to Dr. Hemant, Registrar Administration. And last but not the least, a hearty welcome to all our dignitaries, to all our participants for this program. So, as I said, we are very happy that we are collaborating with the Association of Medical Biochemists of India, and I'm sure this is going to be a very fruitful session for all of you. In the earlier days, genetics did not have any major role, and actually it wasn't even a well-known field. Any lab which was running genetics was considered to be a very high-tech lab, but it's not like that anymore. Genetics has become a part of any lab, and karyotyping is being performed almost everywhere. 20 years ago, karyotyping was the only type of genetic test available, 20 to 25 years ago. But now there are so many advances, there is so much progress in the field of genetics that unless we have FISH, microarray, and now there is even the sequencing, all these are a must, a package for any molecular diagnosis. So that is the reason why we have planned this workshop. I'm sure all of you are going to benefit a lot from this. We will be focusing mainly on karyotyping and FISH, and there will also be a talk on microarray. So it's going to cover almost the entire molecular genetics. So once again, I welcome all of you, and I'm sure you're going to enjoy this session and gain a lot of knowledge from this. We will inaugurate this water. So we will start the program by watering the plant.
Discuss the dignitaries to kindly watch the plant. Everybody. Now, we have request Dr. Megha Rao Madam, our principal and dean from my medical college, to say a few words of wisdom.
Good morning to all of you. At the outset, the group which is putting these efforts to hold this workshop must be congratulated. This appears to be a very timely effort on the part of the organizers. And more than that, it is a collaborative effort which is going to reap the benefits for the delegates. So I must congratulate the Medical Biochemists Association members who are participating in this, in organizing this event, and the members of our Central Research Laboratory, Ramaiah Medical College, led by Dr. Vijaya Mysurkar. And Angel Bula also to be, supporting her all the time. So let's all, on this note of, we are so delighted about this collaborative effort. Dr. Vijaya has already stressed the need, which is almost an unmet need. Today's requirement, as she rightly pointed out, is a lot about the genetic workup in a given case. Lots is spoken about what is termed as personalized medicine. And everything in personalized medicine revolves around the genetic workup in a given case. It's also remarked to an extent that genetic study in a clinical condition is now a part of the routine prescription in not one odd disease entity, but almost across the board in most malignancies in particular. There's also of great importance for the clinician when we are trying to look at the potential for suffering from adverse drug responses, which could be idiosyncratic And a lot of times idiosyncratic reactions are fatal. So this genetic study is now going to become and attain a great importance. And this is considered by clinicians now. Genetics are now constituting the clinicians' effective care teams. That said, is a great profound need to go into the details. I am not anywhere close to genetics as far as my knowledge goes, because we are still around what we learned decades ago, which is exactly what was pointed out by Dr. Vijaya. She said earlier it must have been only karyotyping. Today, it is not just that, it has been elevated to sequencing. And there are lots of terms which I will not attempt to use for fear of being wrong in it. So I leave that prerogative to all the experts who are here, the speakers who are going to be deliberating on the essentials during this workshop. And that said, it's very heartening to know that you have so many delegates participating in this kind of a workshop. Of course, we don't expect some hundreds of them. There will soon be a time when even that will happen, because the way the personalized medicine is gaining importance in clinical practice. And that is why the importance for genetic studies is also escalating by the day. On this note of congratulations and wishing all of you a good, useful takeaway at the end of the day. Good luck to all of you. Have a nice workshop. Thank you very much.
Good morning, everybody. I take this privilege for me to propose the vote of thanks on this occasion. On behalf of the organizing team, I thank our beloved principal madam and team, Dr. Medha Rao madam, who's in despite of her busy schedule, have found time to grace this occasion, and also thank her, and also I thank her for the kind words and encouragement. She has always been a great support to us. Thank you very much, madam. I also express my heartfelt thanks to our Vice Principal, Dr. Shalini Moin madam, for her support to conduct this workshop. Thank you. I thank Dr. Harish sir, Vice Principal and Associate Dean, who has not been able to join us, but I also thank him for all the support that he's been rendered to us, and he's been a great support always. I thank our Registrar, Dr. Pratap sir, for all the support he has rendered to us whenever we have needed. Thank you very much, sir. I also thank our Registrar too, Hemant sir, who has been very kind enough to do the required arrangement whenever we have asked for. Thank you very much, sir. I also thank our HOD, Pathology, Department of Pathology, our Ramay Medical College, Dr. Mangla madam, for having been here, accepting our occasion. Thank you very much, ma'am. My heartfelt thanks to Dr. Vijaya Maisurkar madam, Senior Professor, Pathology and Head of Department, Head of the Central Research Laboratory, for all the support and guidance throughout this workshop. Thank you, ma'am. I thank all the participants of the workshop for having come here. I'm sure this workshop will be a great help to you all. I owe my special gratitude to all the CRL team, Mr. Jaykumar, Ms. Trisha, Shivlingaya, and all the non-teaching staffs as well. They have worked very hard to ensure this occasion, that this occasion becomes a success. I thank everybody, all our team. My special thanks to Ravi and the IT team for the immediate support and the arrangement to be done at the required time, and they have conduct this program. Thank you, the IT team. Last but not the least, I thank all the faculties and the invitees present here for accepting an invitation and having come here. Thank you once again, and thank you all, and I ask your presence to be there for the high tea after this occasion. And join us back after the high tea, back into the same venue for the follow-up talks. Thank you one and all. Thank you once again.
Yes. Thank you so much for receiving it.
Please comfortably, yes, whatever you wanted.
Thank you very much.
So it's a day-long workshop tomorrow. And today also in the afternoon. Okay, so it must be very nice collaboration. That maybe this is the first time we're doing it. I guess we can plan more workshops. If you want the secondary.
Yes.
Okay.
Yeah, the software provided.
Which document is this?
Yes.
Thank you.
Hello.
Thank you. Thank you for being here. Thank you.
ಇಲ್ಲ ಅದು ಇಷ್ಟಪಡ್ತಾರೆ
ಇಲ್ಲ
ಬ್ಯಾಂಕ್ಗೆ ಹೋಗ್ಬೇಕಂದ್ರೆ ನೀವು ಇದನ್ನ ಪಕ್ಕದ ಮಾತಾಡೋದು
ಜಾಯಿನ್ ಆಗಿ
ನೀನ್ ಮೊಬೈಲ್ ಮೇಲೆ ಜಿಯೋ ಟ್ಯಾಗ್ ತಗೊಂಡ್ಬಿಡ್ಲಿಕ್ಕಾಗತ್ತಾ? ಅದ್ರಲ್ಲಿ ಯಾರ ಮೊಬೈಲ್ ಮೇಲೆ ಬರುತ್ತೆ? ಯಾರ ಹತ್ರ ಇದೆ ಅದು? ಇನ್ನು ಯಾರಿಗೂ ಇಲ್ವಾ? Just check and take it off because ಅವ್ರಿಲ್ಲ ಅಂತ ಜಿಯೋ ಟ್ಯಾಗ್ ಇನ್ನೊಮ್ಮೆ ಮಾಡ್ಲಿಕ್ಕಾಗಲ್ಲ. ಇನ್ನೇನಾದ್ರೂ ಆಮೇಲೆ ಮಾಡ್ಬೋದು. ಇಲ್ಲ
You got biscuits or take one of you. Are you there? Let's see, huh? Because so it's some different places, you know, all in various places. But only got what also is here only. There was, yeah.
They got their removal thing, you know.
It's terrible, you know. Oh. Overall. What is that it says? Say it.
Welcome.
I put mine. I opened also, but it's not coming on the screen.
Oh, it will come. This is enough now.
No problem. Screen should be done. Changing this.
Same thing. This thing. First, so sorry. Next back and forward.
No,
Change.
no, let it be.
No, once you
Put it to
change.
slideshow. Not changing. No, you have not put it to slideshow, no?
Oh, now you change.
Right?
1 second. I'll change. I mean, now you change.
Why it is not changing? I'm doing this middle button only, correct? No?
Yeah.
Middle button only.
Same, we use this in the home.
Oh, it's not changing.
हाँ सर, हमारे campus. Council too. Council room or council hall? I'll ask the passport. I'll ask and I'll tell you.
Oh, yeah.
The QR code is not camera.
Oh, which sir?
Camera control.
Is it okay?
1 second.
Don't we have to stay? We have to sit there and wait.
No.
Or you give me the laptop there and change over there. No, if he puts the laptop on that. Podium.
Council hall, the password I know. Sorry. Yeah, password.
Changing.
I'll get the other one. Get me 2 minutes. I'll go to the hall and come back.
Or if it can be changed, you can keep the laptop.
I'll get you.
Connected now.
I'll be connected.
Connections with. Completely reconnected.
Okay, then quickly wait. Eleven it is already.
10:30. 10:30, I think.
Eh, no, what's the time?
55 months. 10:55.
10:55. You have a class at 12.
When you finish by then. Where we go, you know, but
Yeah. Top floor, the top price. Geotag ಆಯ್ತಾ? Geotag photo?
inauguration ದು ಏನಿಲ್ಲ ಅಲ್ಲ.
Inauguration geotag ಆಗಲಿಲ್ಲ.
workshop ಅಲ್ಲಿ ಯಾಕೆ ಬಂದ್ಮೇಲೆ?
ಅದಕ್ಕೆ ಒಂದು geotag ಆ ಸ್ಕ್ರೀನ್ ಒಂದು geotag ಹಾಕ್ಬೋದು. ಲೈಟ್ ಆಗಿ ಲೈಟ್.
ಇಲ್ಲಿ Google Meet
There is a clock. The clock is 5 minutes late. Everybody came from the intelligence of the It's not working. It's not changing only. Slide is not changing only. Changing.
Yeah.
Okay. Good. It's working, no? All lights are switched. All lights are off.
You want all the lectures? Okay, I'll switch off.
One light we can keep, 1 or 2 lights, but all is too much.
It's
Within 1.
Sorry for the slight delay. So we begin today's workshop with an introductory talk on cytogenetics. So I'll just be introducing you to the topic of cytogenetics.
You press one.
So all of you must be knowing this, of course. This is a cell. This is a cell. It has the nucleus, the cytoplasm, and of course in the cytoplasm all these, and then we have the endoplasmic reticulum, the lysosomes, ribosomes, everything. But what we are concerned today with is this nucleus, and in the nucleus, what we are worried about today is these chromosomes. We are talking about these chromosomes which are inside the nucleus. So this is the nucleus with the nucleolus, and here are the chromosomes. So this is what we are focusing on in today's session. So just a brief history. Of course, this study on karyotyping and chromosomes began much before 1960, but the significant achievements which have taken place, the significant landmarks are in 1960, the Denver conference, where the nomenclature was proposed, and that nomenclature is being adopted even now. In 1963, in the London conference, official sanction of the 7 groups, which we follow till today. This official sanction of the 7 groups was by Lionel S. Penrose. In 1971, Kasperson et al. Showed us the first banded human karyotype, G banding, which is also being adopted till today. In 1971, in the Paris conference, chromosomes, regions, and bands were defined. And then the International System for Human Cytogenetic Nomenclature, ISCN, is being upgraded every year. It is every few years it's being revised. So the latest revision was in 2020. So the chemical composition of chromosomes. Now this is the chromosome. And as I showed you, it is situated inside the nucleus of the cell. So this chromosome is composed of DNA, basic histones, neutral proteins, acid proteins, RNA, and polysaccharides. So basically, this is the composition of a chromosome. So it has a large amount of DNA along with other proteins like histones, neutral proteins, acid proteins, RNA and polysaccharides. And this is the shape of the chromosome, which we'll be discussing a little more in detail. Then coming to the structure of chromosomes. Now this is the structure of a shape of a chromosome. It has a centromere and it has a short arm. Each chromosome has a short arm and a long arm. Actually, the chromosome is composed of two chromatids, which are joined together at the centromere. So actually, the chromosome has an X shape. But when we see it in when we do karyotype, we do not see these two chromatids as separate. We just see it as one long thread-like structure. But actually, the chromosome is composed of two chromatids which are joined together at the centromere. So this is one chromatid, this is the other chromatid, this is the centromere. And the tips of these chromatids are called the telomeres. So there are telomeres here. And then there is a short arm and a long arm. So depending on the location of the centromere, the long arm and the short arm will have, the size may vary. That also we'll be coming to now. So this is again another chromosome. This has a slight difference in the morphology in that you can see that there is no definite short arm over here, like I showed you in the previous picture. So there is only a long arm. The short arm is extremely small, that it is hardly made out. There is a short arm, but it's extremely small. And instead, there are satellites, two satellites here with a stalk. So this is the structure of some chromosomes like 13, 14, 15. Some of the chromosomes have this kind of a shape. So instead of having a P arm, instead of having a well made out P arm, there is a small P arm, a very small P arm, a Q arm, and instead of the P arm, we have satellites connected by a stalk. So that is the structure of some of the chromosomes. Then coming to the classification of chromosomes. Now, based on the location of the centromere, chromosomes can be classified, have been classified into three types: metacentric, submetacentric, and acrocentric. So when the centromere is exactly in the center of the chromosome, that means the P arm is equal to the Q arm in length. That is known as metacentric chromosome. For example, chromosome 1, chromosome 3, some of the chromosomes are metacentric. So the centromere is exactly in the center. Submetacentric means the centromere is at closer to one end. So the P arm is really small and the Q arm is really long. P arm actually means the short arm. Q is the long arm. P petit, that is how the letter P was. Petit means small. So that is how the P has been derived. So the P arm is actually shorter than the Q arm. That is submetacentric chromosome. And as I showed you the earlier picture, acrocentric chromosomes have an extremely short P arm. They have a Q arm, and instead they have satellites which are connected by small stalks. So these are three types of chromosomes. Some of the chromosomes are metacentric, some are submetacentric, and some are acrocentric. So again the same thing. Now this is the actual chromosome picture which is here. So chromosome 1 metacentric here. You can see the centromere here. This is diagrammatic. This is the actual chromosome. This is submetacentric. The P arm is shorter than the Q. And this is acrocentric where there are satellites. And this is the Q arm. P arm is extremely small. So 22 pairs of autosomes are there and a single pair of sex chromosomes. XX in females, XY in males. This is known as the diploid number. And the members of a pair are called homologues. So there are a pair of chromosome 1, pair of chromosome 2. So the two members are called homologues. In males, it is 46XY, in females, 46XX. And the gametes are always haploid because when the gametes unite together, then we will get the diploid set. Then euchromatin contains genes which are actively expressed. So in every chromosome, there is euchromatin and heterochromatin. Euchromatin contains the genes which are actively expressed. Heterochromatin has the inactive, unexpressed, repetitive DNA. So here, if you see it in this picture, the orange colored are the heterochromatin, and then this is the euchromatin. This is the centromere. These are the telomeres. So the telomeres are actually heterochromatin. Then coming to the classification of chromosomes, there are 7 groups of chromosomes, A to G, based on the size and the position of the centromere. So group A consists of chromosomes 1 to 3. These are large metacentric or submetacentric, as in chromosome 2. Group B has chromosomes 4 and 5, large submetacentric chromosomes. Group C has chromosomes 6 to 12 and X, medium size submetacentric. Group D has chromosomes 13 to 15, medium size acrocentric with satellites, as I showed you. Group E is chromosomes 16 to 18. These are shorter meta or submetacentric. So they are submetacentric but shorter than the group the C. So group E chromosomes are shorter than the group C chromosomes. Group F has the shortest metacentric, chromosomes 19 and 20. And group G has chromosomes 21, 22 and Y. These are short acrocentric with satellites. Y does not have satellites, 21 and 22 have satellites. So all of you must be familiar. Are you familiar with this classification of chromosomes? Or are you hearing it for the first time? You're hearing it for the first time? Okay, so now have you understood it? This is the classification which has always been adopted. Whenever we study chromosomes, we always go by this grouping of chromosomes. So this is the what I showed you earlier. So you can see here, we always when we do a karyotype, conventionally we have to group the chromosomes, just like the groups which I showed you, A, B, C. We don't write A, B, C, but this is these 3 are in group A and they have to be put together. Then after a short gap, small gap, groups B, 4 and 5. These are all group C, D, E. So that this is how we group the chromosomes, and conventionally always the sex chromosomes have to be put last. Right down. Okay, so this is how, and again you can appreciate that this is a long metacentric chromosome. These are submetacentric chromosomes. These are acrocentric. Can you all appreciate now? Now I've told you the classification. Can you appreciate this? This of course is going to be again shown to you when you study the when we demonstrate the karyotyping. So now I have you understood this is known as a karyotype. Are you clear about this now? And the different groups? So now what are the indications for cytogenetic study? When do we have to do a cytogenetic study? Whenever the patient is having a known chromosomal syndrome, just for confirmation sometimes. If the person has unexplained mental retardation, usually it will be a child with unexplained mental retardation or a child with delayed milestones. So the parents are worried why the child is not yet walking, why the child cannot stand up, or why the child cannot speak. So delayed milestones, dysmorphic features. That means any obvious abnormality in the face, some definite abnormality in the appearance of the individual. Congenital malformations. A baby born with congenital malformations, many times it could be because of genetic abnormality. Ambiguous genitalia. This is another condition where we have to go for cytogenetic study. And later on in life, sometimes delayed pubertal changes. Nothing may be diagnosed till the age of puberty. Then the girl does not attain menarche at all, or the boy does not show any secondary sexual characteristics. Then we will go for chromosomal study. Indications for these are the indications for cytogenetic study. And another indication is infertility, recurrent spontaneous abortions or stillbirth. These also could be because of genetic abnormality. Then at risk, pregnancies which are at risk. So when do we say that a pregnancy is at risk? If there are certain serum screening tests which are indicative of risk, or if the scan is showing, ultrasound, the prenatal scan is showing some abnormality, or if there's a previous abnormal child, then also we say that it's a pregnancy at risk. Because again, we don't want the next child also to be abnormal. So previous abnormal child or even maternal age more than 34 years is also considered as pregnancy at risk. So all these are also indications for cytogenetic study. If the parents are known carriers of a mutant gene, so the parents may sometimes approach the geneticist and say that one of us or maybe even both of them may be carrying some abnormality like maybe sickle cell trait or cystic fibrosis gene. So if one of the parents is having even one abnormal gene, they are worried that they will be transmitting it to the offspring. So then they may want a genetic study. So that genetic study may have to be carried out on the fetus. So this is another indication for cytogenetic study. And cancers like leukemias, soft tissue sarcomas. So even in cancers, we do genetic study for typing of the cancers. So this is a large number of conditions where genetic study is indicated. So what are the samples that we take for chromosomal analysis for karyotyping? Peripheral blood lymphocytes, bone marrow, of course, depending on the indication. Peripheral blood lymphocytes in most of the cases, which I told you. Bone marrow in leukemias, skin fibroblasts sometimes, umbilical cord blood, if you want to study the in the fetus. Then the amniotic fluid cells also, if you want to study the, when you want to do a genetic study on the fetus. Chorionic villi, tumor tissue, if it is a cancer case. So these are the different samples that we can use for chromosomal analysis for karyotyping. Now this is G banding, which is this is going to be shown to you more in detail. So this is the technique that we usually use, and this is known as a metaphase. Now this whole cluster of chromosomes, if you count, there will be exactly 46 if it's a normal individual. These are all the chromosomes from one nucleus. These chromosomes were all in one nucleus, and the technique will be explained to you by Dr. Angel in the form of a video. So after we do the processing, where all these chromosomes have been released from one nucleus, this is known as a metaphase. And then we stain by gene sustain, that also will be shown to you. And that technique is known as G banding. So what I want to stress on now is the dark bands and the light bands. When we do G banding, each chromosome shows unique pattern of dark bands and light bands. So dark bands correspond to the adenine thymine rich DNA. And the light bands, can you see here? Like a zebra, no? Can you see here stripes on the chromosomes? We don't call them stripes, we call them bands. So the dark bands are adenine thymine rich DNA. The light bands are the guanine cytosine rich DNA. So, and how do we make use of these dark and light bands? It is for identification of the chromosome. Each chromosome has its unique banding pattern. So chromosome 1 and chromosome 2 will not show similar banding, they show different banding. And that is how we can identify as chromosome 1 based on the size of the length of the chromosome and the binding pattern. So that is how we are able to precisely identify each chromosome from 1 to 22 and X and Y. Okay, so have you understood? Each chromosome has its unique banding pattern. And those bands are displayed by a technique known as G banding. This G banding will be explained to you by Dr. Angel. And then finally, that was a metaphase which I showed you. All the chromosomes were scattered. And then with the help of software and of course with by the person's knowledge about the identification of the different chromosomes, finally you can see that the chromosomes have been arranged. Do you understand? In the metaphase, the chromosomes were all scattered. Chromosome 1 must have been next to chromosome 5, and then all the chromosomes are just in a bunch. You cannot make out, you cannot identify, and you cannot make out whether there's an additional chromosome, whether there's an abnormality. So we need to arrange the chromosomes before we detect any abnormalities. So for that, now this arrangement is done with the help of software and by the geneticists to identifying each chromosome. So you can see the chromosomes have now been paired and put. And as I told you, this is the A group, B group. This is how we have to arrange the chromosomes in the same way. We cannot put them, this gap has to be there. Here also there has to be a gap that is to delineate the different groups. And you can see that each chromosome has its own banding pattern. Can you appreciate that? No two chromosomes, of course, chromosome 1 and the two pairs in chromosome 1, the two homologs, I told you, homologs will have same banding, but 1 and 2 have different appearance, 3 is different, all the chromosomes have a different appearance. And based on the length, descending order of the length. So can you appreciate that? In the descending order of the length, there we have arranged the chromosomes. So the longest chromosomes are here on top, the shortest chromosomes come down, and as I said, the sex chromosomes have to come last. So X and Y, so this is a male. And the karyotype has to be written as 46, that is total number of chromosomes, comma, capital X, capital Y. If it's a female, 46, capital X, capital X. Okay, clear about this? Any questions up till now? Since you are, some of you are not familiar with this, if there are any questions, you can ask. Because this is what you are going to be doing for the next 1.5 days. And this is the software. Of course, different types of software are available. So this is an example, the microscope, and then this is the computer screen on which these are metabases, and then the karyotype is prepared. There are other banding methods. This I will not go into much detail because nowadays people are not doing these different banding methods. It's become quite obsolete. One is the reverse banding, R banding. So here in R banding, because we treat with heat, the light and dark bands are reversed. So what was the dark band with G banding becomes the light band with R banding. That's why it's called reverse banding. Then there is Q banding, quinacridine banding, where we do fluorescence staining instead of giemsa staining. Fluorescence staining with quinacridine mustard or quinacridine dihydrochloride. So with that, the Y chromosomes and the satellites can be well seen. Then there is C banding, centromeric heterochromatin banding, where the centromeres are well delineated. That is done with heat or alkali denaturation. Then there is high resolution banding. High resolution banding means those bands which I showed you were not many. But if you want to see more bands, to increase the number of bands that are seen, up to 800 bands can be seen per haploid set. So for that, we do high resolution banding sometimes. So here the technique is to inhibit cell division with methotrexate or thymidine, then release the cells into mitosis by adding folic acid, and then add colchicine. So the technique will be explained to you. High resolution banding is a slight variation of the usual technique, that is to demonstrate more bands. Then, so now this was all about the normal chromosomes, the structure. So now just a brief introduction about the types of genetic abnormalities. So genetic abnormalities are basically due to mutation. Mutation, in short, can be defined as a permanent change in the DNA. So there are three broad categories of genetic abnormalities. The first is the genome mutation. In genome mutation, what happens is an entire chromosome is lost or gained. So abnormal number of chromosomes. Monosomy is when a chromosome is lost. Trisomy is when a chromosome is gained. For example, the chromosome 21. If an additional chromosome 21 is present in the individual, then what will be the total number of chromosomes? Obviously, it will not be 46, it will be 47. Because the chromosome 21 is instead of one pair, that is instead of two chromosomes 21, there are three. So that is known as a trisomy. So the total number of chromosomes itself will change. Either it will be less or it will be more. So either less than 46 or more than 46. That is called genome mutation. Chromosome mutation means a structural change. So the total number of chromosomes is the same, normal, 46 only, but part of the genetic material from 1 or 2 chromosomes or sometimes 3 chromosomes, part of the genetic material is either lost or goes to another chromosome, that I'll be telling you. So part of the genetic material is altered, but the total number of chromosomes remains the same. That is a chromosome mutation, structural change. And gene mutation means at a still lower level. Now all of you know that on the chromosome there are multiple genes, large number of genes on each chromosome. So just one single gene is altered. Even alteration of a single gene will give rise to disease. Alteration of a single gene is a gene mutation. It could be either a point mutation or a frame shift mutation, which I'll be showing you. So these are the three broad categories of genetic abnormalities. Genome mutation, that is abnormality in chromosomal number. Chromosome mutation is abnormality in chromosomal structure. And gene mutation is a single gene is altered. So in the first group, the total number of chromosomes is changed, either less or more. In the second and third groups, the total number of chromosomes remains the same. All of you understand this? Any doubts in this?
Just a minute. Just a minute.
So I told you about point mutation and frame shift mutation. In point mutation, one single nucleotide is changed. only one single nucleotide. In the DNA, you know, no, there are multiple nucleotides. So when one single nucleotide is altered, then it may be replaced by a different nucleotide. That is a point mutation. Frame shift mutation means, then I'll show you the diagram. Yeah, this is the point mutation. See, for example, this adenine has changed to cytosine. Just one nucleotide has got changed, and that has changed the whole DNA. It has become into a different structure altogether. So that is a point mutation. In frame shift mutation, there is deletion or insertion in a DNA sequence. Now here, this is the DNA sequence which was there. GAT, CGT, GAT. So here, this has got added. See here, can you see this? The sequence is GAT, CGT. Here, GAT, CGT are here, but in between, valine has been added. So there is addition or insertion of valine. So what has happened? The entire nucleic acid formed is changed. So now it has become into arginine, methionine, tyrosine. So just addition or deletion of one DNA sequence or one nucleotide has shifted the way that the sequence is read. That's why it's known as frame shift mutation. So here it was leucine, alanine, cysteine. So, and here it has changed to arginine, methionine. So because of deletion or insertion of in a DNA sequence, the whole sequence has changed. Clear about this? Point mutation is replacement of one of the bases, and here it is addition or insertion. So then, these were what I told you was about the single gene defects. Now they're coming to the abnormalities in chromosomal structure. I just quickly go through the different types of abnormalities in chromosomal structure. So one is balanced reciprocal translocation. Balanced reciprocal translocation means, now here for example, this chromosome 4 and 20. Green is chromosome 4, red is 20. What has happened here? A small part of the chromosome 20, red part has come to the green chromosome 4, and the small green part has gone to chromosome 20. So it is like sharing of material. From one chromosome it goes to the other, and from this one to the first chromosome. So it is just that the genetic material is just shared between the two chromosomes, or its position is just changed. So there is no change in the number of chromosomes. It's just that there is rearrangement of the genetic material, but of course this can lead to it could lead to changes in the phenotype of the individual. So clear about balanced reciprocal translocation. Why we say balanced is because there is no loss of genetic material. There is no loss at all. It is like you take money from one pocket and put it to the other pocket. There is no loss of money. In the same way, genetic material from one chromosome goes to the other, and from this to the first. There is no loss of genetic material. That is one type of abnormality. So this is the same thing which is shown in this form. A, B, C, D, E, H, I, J have gone to this chromosome, and N has come to the first chromosome. So some of the genes from one chromosome go to the other. Then Robertsonian translocation is another type of translocation. This involves acrocentric chromosomes. These are chromosomes 14, 21, two acrocentric chromosomes. What happens here is that the P arms break. For one of the chromosome 14 will lose the P arm. Similarly, one chromosome 21 also will, the P arm will get broken, and then these two P arms join together, and the Q arms join together. So what has happened? Now this is one chromosome 14 which is normal. The second chromosome 14, the Q arm has joined with the Q arm of chromosome 21. This is the normal 21, and these are the two joined P arms. So we get one unusually long chromosome, 14Q, 21Q, and a very short chromosome, 14P, 21P. This usually gets lost at the time of subsequent cell divisions. This is known as Robertsonian translocation. It's also known as centric fusion, because fusion occurs at the centromere. Here also there is not much loss of genetic material. A small amount of genetic material gets lost. The two fused P arms usually get lost at the time of subsequent cell divisions. Then deletion is another type of structural abnormality that is very easy to understand. A small part of the chromosome breaks off and gets lost, gets deleted. So this chromosome, which was so long, has become a little shorter because of the deletion. The deletion could be at the end of the chromosome, terminal deletion, or it could be in the middle, that is interstitial deletion. So this is the same thing in different depicted in this way. Inversion. Inversion is when there's a breakage in the chromosome. So this is the centromere. In one arm, you can see that there's a break. And what has happened? That broken part has got reversed and got attached in this way, reverse way. So this green has gone on top and the red is down. Here the red was on top and the green is down. So this is called paracentric inversion when the break is on the same side of the centromere. Sometimes the break can occur on either side of the centromere. That means the centromere also is included in the breakage. And the centromere also will rotate, get inverted. This is and reinserted like this. This is called pericentric inversion. Clear about this? Paracentric is in one arm only there is a breakage, paracentric. And if the breakage is on either side of the centromere, it's called pericentric. So that is inversion. Then insertion is just addition of a small bit of genetic material inserted into the chromosome. Duplication is when there is some duplication of genetic material. You can see that this portion has got duplicated in the chromosome, that is duplication. These are all different types of genetic abnormalities, structural abnormalities. Then isochromosome. Isochromosome is one type of structural abnormality where one entire arm gets lost and this the other remaining arm only gets duplicated. So it's like mirror image. Can you see here? This blue was here, but this whole arm has gone. And here, this has got formed a mirror image. So that blue, actually, this much of genetic material has gone off, it's disappeared. So it could be either loss of the P arm or the Q arm. So loss of one arm and duplication of the other. So this arm is lost, and instead this arm has got duplicated. This becomes like a mirror image. That is isochromosome. And then ring chromosome is when this chromosome, like here, it has, you can see that both the tips have broken off. When the tips of the chromosome break off, the ends become very sticky, these two ends. So the chromosome automatically curls up and becomes like a ring. These two broken ends come together and join. They get attracted to each other and this is a ring, chromosome like a ring. So these are all different structural abnormalities in chromosomes. Then lastly, we'll come to the nomenclature of chromosomes. This is the International System for Human Cytogenetic Nomenclature. ISCN, this is what is being adopted regularly. So according to this nomenclature, there are certain rules which we have to follow. So chromosomes, as I showed you, 1 to 22 have to be arranged as pairs. Sex chromosomes X and Y have to come last. The arrangement has to be in decreasing order of length. Then P arm always has to be upwards. The short arm has to be up and the long arm down. We cannot reverse it. So P arm upwards, Q arm downwards, and karyotype has to be written in this way, as I told you. 46, X6 or 46, XY. X should be capital. Y also should be capital. So this I already showed you. This is how the karyotype has to be prepared. And now again in that there are certain other rules also to be followed. So we now these are certain landmarks in the chromosome. The centromere, the end of each arm, and the most striking bands. By making use of these landmarks, we can further divide the chromosome into different regions. I'll show you. So first to keep in mind these landmarks. The centromere is one landmark. The ends of each arm are landmarks, and the most striking of the bands, these very dark white bands. So now how do we divide the chromosome? Each arm of a chromosome is divided into regions. The area between two adjacent landmarks. Each region is divided into bands. So the bands may appear darker or lighter staining. Each band is further divided into subbands, and the numbering is done from the centromere. This may look a bit confusing. I'll show it to you in a diagrammatic form. numbering should be done from outwards. So I'll show it to you in the diagrammatic form. Now this is how it is. This is the P arm. In the P arm, the region between the portion between two landmarks is called a region. So this is region 1. Then this is region 2, region 3. Similarly, in the Q arm also, region 1, 2, 3, and 4. Different regions have been marked out. Then in the regions, bands and subbands have been numbered. So region 1, the band number, the first band is 1, 1. Second band will be 1, 2. Then for region 2, the first band will be 2, 1. Second will be 2, 2. So like that, bands and subbands are delineated. I'll show you that. Like this. Now this will help you to understand it easily. So how do we designate the bands and subbands? Now suppose we say, suppose we denote one particular region. Now why this is important is, sometimes the abnormal gene will be located at that, if the abnormal gene is located at that point, how do we explain that point to somebody else? Internationally, everybody should mark out that point in the same way. We cannot have our own way of saying that on the P arm it is located here. We cannot have our own way. We have to have some well-recognized way of denoting it. So this is internationally accepted that always first we put the chromosome number. So chromosome 1. On chromosome 1, second is the arm symbol P. In the P arm, on the P arm, region number 3. I showed you what is region, no? Between two adjacent landmarks is the region. So in region 3, band number 1, then we put a decimal point, and subband number 2. So this is the location of the abnormal gene. So precisely anybody in any country, if you tell them that the abnormal gene is located here, they will be able to find the exact spot. So clear about this? It may appear a little complicated, but clear about this? Because sometimes you may see in textbooks, no, that the abnormal gene has been found to be located on this particular 1P31.2. So what is that actually? It is the exact precise location. First the chromosome number, then the arm, then the region, then the band, followed by decimal point, and then subband. That means in region 3, band 1, subband 2. That's what they mean to say. So this is the internationally accepted nomenclature. Then this is an ideogram. For each chromosome now, they have done this division into bands, subbands. So this helps us when we are doing karyotyping. If we find an abnormality or a breakage, we can exactly locate the breakage with the help of this. This is known as an ideogram or a chromosome map showing the banding pattern of each chromosome. Of course, this is available now inbuilt in the software, and we can easily locate any abnormality. We can tell the precise location of the breakage or the abnormality. So these are various symbols that have to be used. I've already told you most of these. PM, PQ, CEN is centromere, DEL means deletion, DUP is duplication, I is isochromosome, INV is inversion, R is ring, T is translocation, TER means terminal. That is PTER, P terminal or Q terminal. Then when there is mosaicism, that means two different karyotypes in the same individual. Then this is how we have to denote it. These are all internationally accepted ways of denoting. Plus or minus means gain or loss of a chromosome. For example, 47, XY, plus 21. What does that mean? That the patient has a total number of 47 chromosomes because one chromosome has got added, XY means male, plus means gain, and 21 means chromosome 21 has been gained. That is known as Down syndrome, no? Trisomy 21. 46, XX 5P minus means total number is 46. XX means female. 5 means chromosome 5. P on the short arm, there is a loss, deletion. 5P minus. So these are all internationally accepted ways of denoting any abnormality. And then structural changes, always we put a semicolon like this whenever we want to denote structural changes. So this is an example just to show how trisomy 21. See, we can see an additional 21. These are just examples of abnormal karyotypes. Then here there is deletion of 5. You can see it has become short. Here there is translocation. Can you see here? From 9 to 22. This is what I told you as balanced reciprocal translocation. from 9 to 22 and 22 to 9. So this is how we denote it. 46 total number, XX female, T means translocation, 9 to 22 means from chromosome 9 to chromosome 22 and 22 to 9. Q means long arm. These are the region and the band. Then semicolon to show that there is a translocation, and again the region and the band. So where exactly chromosome 9 has broken is here, and where chromosome 22 has broken is this location. So that is why I told you about the regions and the bands. So this is just an example how a karyotype is finally given. This of course will be shown to you in the demonstration. So this is just as an example, the patient's name and all the details, then the karyotype, the metaphase here, and then here we give the final impression. Now, just for completion, this all is going to be told to you. I'm not going to talk about this in detail. So karyotyping, I told you in detail. Other techniques for genetic study are, one is FISH, just an example of trisomy 21. You can see 3 signals. Then here another example of FISH where a chromosome has been painted. This is chromosome 4 painted. Then there's another specialized technique known as spectral karyotyping where we use 5 different fluorochromes and the different chromosomes will take up different colors. And then finally we get a very colorful picture like this. That's why it's called spectral karyotyping. And then based on the colors, we can arrange the chromosomes and form the karyotype. So here we are not using G banding to arrange the chromosomes, but we are using the fluorescent color that the chromosome has taken. This of course has to be done under the fluorescent microscope. So this is the karyotype. This is called spectral karyotyping. Thank you. I hope you have understood all these basics. If there are any questions, you can ask. Any questions?
Any questions? Okay.
There are so many different factors. How would you explain example of somehow?
No. That's what I said. Each chromosome still has its specific binding pattern. So chromosome 6 will have a particular appearance. Chromosome 7 has a different appearance. That actually Dr. Angel will be showing you.
Deletions are all take place in the patterns.
Yeah, it is a little, that of course needs a bit of experience. Correct, I agree, yes. If a small part of one of the chromosomes is deleted, it may start resembling another chromosome. That's true. But with a little experience, it can be done. Even then, if there are problems, then we have to go to other methods. Like that's why karyotyping is the basic test. If still we have some difficulty, then we may have to go for FISH. Of course, depending on the requirement. We may have to go for some additional tests also. The karyotyping, in many cases, we get complete information from karyotyping. Sometimes it may be difficult. So that actually the appearance of each chromosome, Dr. Angel will be showing you when you see the techniques. Each has a different appearance. So nobody can confuse chromosome 6 with 7, 7 with 8. The confusion will not take place. Any other questions?
Any more questions? So we'll go to the next talk. Thank you very much, madam. The next talk actually is by me. We are trying to change the schedule because Mr. Ravi from Thermophilia would like the topic. So I request Mr. Ravi, application scientist from Thermophilia, to give a talk on nitrate.
One sample. Yeah. I took
Very hard. First presentation. Which one? CDC. CDC. Everyone. Can I start? I'm already to the last. What's happening? Good afternoon, everyone. Take a few minutes time to introduce myself. I'm Raju Yadav, product specialist for next generation sequencing and microarray from Thermo Fisher Scientific. So I will go to first slide to discuss on the microarray front. So what is the technology, how the machine looks like, and how it's going to be utilized in diagnostics and research space. If you have any questions, feel free to ask. We can able to discuss on that, and we can move on further to the slides.
Slide.
You.
So Thermo Fisher, as I just said, it's a life science company, so we serve different kinds of life sciences customers, starting from your PCR to electronic microscope, mass specs and everything. So we are also the division called Genomics Instrument, where we supply, we support with standard sequencers, data PCRs, digital PCRs, microarray and detection systems. So what we largely discuss today is going to be sorry for the. Yeah, so this is a microarray system. So probably this we call it Napimetrics. Napimetrics is a very the pioneer in the microarray technology. So we've been serving big industry for a few decades. So this machine is called G-chip scanner 3000. So basically, in a microarray system, we have all these components. So we have the hybridization oven and fluidic station and the scanner, this scanner, and we can upload it. This is the complete component as a package. The total system is called a microarray system. So basically, what is the applications we can able to in this microarray system is, so what is the difference between we can say in other words, the microarray system is an advanced technology than the karyotyping. So if you are already seeing the karyotyping requirements and order for it. So basically the karyotyping you largely do it by the culturing the cells, and you can able to see the your chromosomes come under the microscope, and you will able to detect the deletions or duplication events. But what is the difference in the microarray is the sensitivity curve. So typical karyotyping can able to resolution will be up to 2 MB sizes. So anything, the deletion of the chromosome parts or the portion of or the duplication parts, you can able to have the resolution till 2 MB. So anything found below 2 MB, so you cannot really visualize under the microscope. So, but there are a lot of disease conditions which will impacted or which will caused by the even single base resolution. So even one SN base also can cause some diseases, right? So all these diseases, when you're trying to do with the traditional karyotyping way, so this information will be missing in the karyotyping. And also it requires culturing, a lot of culturing you have to do when you do the karyotyping, and it can also lead to the possibility of contamination so that you cannot get the desired results. So and also the diagnostics yield of the karyotyping is compared to less because the limitation of the resolution is of approximately 2 MB. So whereas a microarray will beat all these challenges, so where you will get a single base resolution as well. Even if there is a single mutation in the given sample, we can able to get the resolution of this. And there is no culturing required. You can directly isolate the DNA from your amniotic sample or product of conception or any kind of samples, even also can take the samples and you can pretty much analyze with the microarray technology. So the turnaround time is also very less compared to karyotyping. 4 days is the turnaround time for you to sample to report. So the brief advantages of the karyotyping, to put it more briefer, is you have more resolution. Single SMP resolution has. The turnaround time is 4 days comparatively, and the diagnostics yield is almost close to 30% extra than what you get from the traditional karyotyping now. So these are the key differences when you really talk about the karyotyping. And also the microarray technology is recommended as a first line recommendation from the American College of Organics. And also it's been probably put in use. So there's a first line technology right now. And also it is being utilized as a confirmatory tool for the non-invasive prenatal testing we do on NGS. So in the regular next generation sequencing, we do non-invasive prenatal testing, which is probably called as NIPT or NIPS, the screening. So if you get a positive confirmation on the screening test, microarray is only the test you can have the positive confirmation. So yeah. So what are all we have in the cytogenetic suite? So we do have 3 different types of capacity of assays in this. So one is Optima, another is called SAM50 and HD. So basically what is an Optima used for is, as it's mentioned in the slide, it is largely for the prenatal application and miscarriage samples and all. So if there is a miscarriage, the fetal autopsy, the tissue will be taken, and it can be screened for the anomalies. So this can be done in 4 days of time, so that we'll understand is there any aberration genetically within the fetus, or even we can test the mothers and all, so that for the next time they can be a little bit careful about their the pregnancies. So also we have a 750K, which is a high density. This, what is the difference? This numbers denotes is the millions of probes, millions of data points, you can say. 750,000 probes will be on there, whereas Optima is called, another way is called 350K, so 350. So why we require more of the resolution when it's going to the 750K is also serves for the postnatal applications. So when you have any developmental disorders, any this neurological disorders, or even for oncology, we have any kind of areas where you have to look at and diagnosis. So that's you have to use the 750K. Cytoscan HD has even more than 5 million markers, a very, very high density array. So it is used even for hematological malignancies or cancers. So microarray is typically used for all this where you want try to, it's almost like, it's not exactly the replacing the karyotyping technique these days, but definitely it can address many queries which cannot be addressed by the regular karyotyping. So and since it's going to be, is already recommended as the first line testing for cytogenetics. So this technology can be pretty much independent. So sensitivity, when you talk about, if you really look at a little bit of sensitivity requirements where we can basically utilize this, we see the Optima, which is a low resolution array available with us, and the next is 750 and the next is HD. If we see these analytical claims, that's where the differs, right? So if you see the gains and the losses are approximately 2 MB less than 1 MB long, and the mosaicism approximately 2 per 20% you can able to detect. So the 750, because it's a higher density arrays, even the deletions and duplications and all can be even the KB levels, which is certainly not possible in the case of my traditional karyotyping. And if you go to even higher resolutions levels, even for the 10 KB kind of gains and loss also can be detected. So this high resolution technology, SMP level technology, and it's a pretty simple hybridization methodologies. So its process is very, very easy. As it were showing in the, I have shown in the first slide. So this is an automated solution. So this is an automated loader. So the system is totally automated almost. So once you process the samples and put in the predicate station, so basically you're staining all this and all. So this process is totally automated. Even the loading of the chip is everything automated. So with this kind of an automated technology, hybridization technology, within this 4 days of turnaround time kind of. So this will really improve your diagnosis with much more resolution, and it will help diagnosis faster and more accurate, so that the diagnosis will be more. The patient treatment and the care can be improved to a different size. So as I mentioned earlier, also it can be utilized without any cultures. If you have any pure samples, like frozen tissues, also culture or even uncultured setting, chronic villus samples. So different kind of samples you can take. There is no limitation in the sample sizes, and we give all the end-to-end solution right from isolation to even to the TIL analysis. So this is the technology which going to drive the future over the karyotyping. So any questions over this? If you want to compare with the karyotyping, can able to discuss. Any questions? Yeah, please. Yeah. So that's what. So you can do a microarray. The reason being, there you do an immunosynthesis, right? When NAPS is coming a positive, the screening test. So the next level is you have to do an immunosynthesis. So immunosynthesis, again, you take the amniotic out and you have to culture it. If it's getting any possible contamination, you cannot repeat the immunosynthesis again. It's too dangerous for the patient. So here we can use the karyotyping also, and you can use a microarray. So what is the difference between karyotyping will give the limited information for you. So if anything is above, because it's all the person who is a very expert who is sitting in front of the microscope, depending on their experience and all. So it's a manual thing, right? So you have to see under the microscope, depending upon how cultures has been treated, how the morphology, chromosomes, everything is read. They will be detailed, and it is little limited on the scale. Since these are the completely driven by the system, and it's a SNP level, even one base, it's level resolution. So bases I will put in very similar structure if few people are not familiar. So when you are deriving the size of your DNA, so 1,000 bases we call as a 1 kb, and 1,000 kb is called as 1 MB, and 1,000 MB is called 1 GB. The human size DNA is 3.2 GB in size approximately. So when it is about 2 MB, the deletion or duplication is largely 2 MB. It can be visualized, and the person who is looking at the technician or doctor can able to report it. Because of this 2 MB deletion or because of 2 MB addition in this particular chromosome, there is a miscarriage that happened. There is a genetic abnormality is happening. What if it is falling below to that? If it is below to 2 MB or 1 KB or even 1 SNP, a single base. So there is no other technology can able to predict with accuracy than this method. So in those cases, your diagnosis yield will be better. If you do a karyotyping, if less than that, we can say there is nothing wrong in this. Karyotyping absolutely looks perfect. So you can go ahead. So if you continue the pregnancy, then when the time the baby is born, then we can see there is some kind of a chromosome aberration in the new baby. So we're using this kind of a technologies, even at SNP level resolution can possible. So the diagnosis will be improved, and we can accurately say, yes, this is the problem, and this is the genetic aberration caused, and what kind of a disease it can be caused. So this full information we can able to give. So this is the confirmatory test than the karyotyping.
I have some questions.
Yes.
As participant asked for NIPT and whether we have to do microarray. I have a question for prenatal diagnosis. Do you recommend 350K or 750K?
So if it is a prenatal, largely for us in Indian population, if we say it looks largely trisomy 21, 18, and 13. So these are the regular trisomies we see it. In those cases, largely we can use an optimum, which is a little lower resolution. To understand there's a bigger chromosome deletion sequence and all, so that is absolutely fine. If you have too much of recurrent miscarriages and again it's coming along, then we have to look at a little bit more deeper insights. We require more data. In that case, we can offer the next level high density arrays so that you will get more information. So, and if you see this optimum, it's not only the losses and gains, we also has 396 special genes which is related to the prenatal pregnancies. So this will give much more resolution. For prenatal, we largely asked to use the low resolution one. But if the recurrent miscarriage, they can certainly use the 750K. So NIPT, what is the difference between these two? NIPT is a screening tool. It's not a confirmatory test. So NIPT or non-invasive prenatal screening, we call as generally we should not call as a test. It's basically a screening tool. So we cannot take it as a confirmatory and take a clinical action on that. So when we get a positive, we have to do a microarray to have the confirmatory. So then only we can take a call. The clinician can further decide the clinical course of action.
What would be the cost of test?
So in the market, this one, the low density one will cost you probably around 12,000 to 13,000 rupees to cost the patient. So, but it looks more when compared to the karyotyping probably 3,000, 4,000, 5,000 rupees. But we also have to look at the amount of clinical data we're getting. The diagnostic yield also we have to look at. And in one shot, it largely don't go wrong. So we get more data, more benefit. So slightly price also will be increased. Because these investments are also is definitely higher than the regular carrot.
Are there like different panels of what do you say tests that can be done just like we have profiles? Do we have different profiles of genes? Because if you are looking for a certain disorder or maybe group of disorders, you want to select a certain panel of microarray. Is it like that, or is it like a general panel that is available with specific probes? How are the
largely when you do microarray, the advantages you can look at the complete chromosome perspective, whole genome perspective. If you really wanted very specifically target like one gene, two genes, I want to look at the group of disorders, the cardiovascular disease or neurological disease or endocrinology disease, something. If you want to really group, but I'm looking on one gene, two genes, three genes, very specific, known conditions and everything. Microarray is not a very right option at that point of time. For you, NGS would be a perfect option. So any targeted sequencing with a perfect array. So protein, what will happen is, so genomics labs, when there is a service provider, genomics labs and all is there, they typically will have microarray on NGS or also Sanger sequencing. And there is another sequencing technology, Sanger sequencing. So that even one gene, even one target, even multiple targets, panels and everything can be completed. So largely microarray is not a very targeted for one or two genes. It will give the complete, the big picture or the complete chromosome level. I mean, complete genome of the level.
So the first would be that once you get a result, right? So you will get to know as to what SNPs are there. And then, so what level of expertise does one need to take what comes out of your muscle and then correlate with the database, or does has a database where you correlate with these SNPs and then come out with
So we have an analysis software called Chromosome Analysis Suite. So it's a complete graphical visualization where we get all this graphical aspect. Even how we see the karyotype. When you see the karyotyping, how here all the chromosomes are written out. Even that visualization is given. So it's already validated globally, and we have in-house validations. And within India, there are almost like 25 to 30,000 samples crossing in India on this platform. So we have a complete use database.
Yeah, so your suite would be an add-on to this machine.
It's an add-on. So we give the free of cost for that. So that the data what is produced by the microarray system can be loaded into that suite, and it can give the results. So it will show all this copy number variations, SNPs, where it's gone wrong, all this information clearly given in form of reports. When we are delivering the report, we have to interpret with the correlated with the clinical correlation. So it's an end-to-end service.
what are the other than the fact that it's being used for the confirmation of non-invasive testing? Any other practical applications it's being put to use other than the research aspect, the diagnostic aspect or therapeutic aspect? This microarrays are being put to use in India.
So this is for diagnostics.
I mean, I'm asking specific, like a group of disorders or something.
Okay, can you repeat the question?
He told us that it is the next line of testing once we have some kind of positive results in non-invasive testing, clinical testing. Other than that, are there any other group of disorders for which microarrays being used currently in India or any part of the world?
Yeah,
practical applications.
Correct. So practical.
I know there are a lot of research-based applications. Just asking, is there any?
So typically microarray where we have is largely for diagnostics use, not for the regular research use much. So it can able to do the transcriptome, gene expression profiling and all. So this particular model and this solution, which is called Cytogenetics solution, is being only for the diagnostics purpose. So where it can be used, so NIPT is one thing, is a confirmatory you use this. That's where you can use this technology. Besides NIPT, it doesn't have its own diagnostics applications. For example, we take the 750 guideline. This is not only used for the regular confirmatory of NIPT. It is also used as a standalone test for the people, for the infants, newborn, postnatal kids, or even adults. Not in adults, but pretty much say in the adults. So they have many developmental disorders. Those can be screened, and also this has another clinical application in cancers. So I didn't have a slide of showing the cancers because largely it's called OncoScan CNSA. So for the cancer, it can use all these copy number variations and all we can predict. So this can be not only as a confirmatory tool for NIPT, it's only NIPT confirmation is only part of it, but as a standalone, it can be used for various tests. But what is the difference of this microarray from the regular NGS? Microarray is a holistic approach. It's a total gamete you need, complete the genome you will be able to study. Whereas NGS can able to do the targeted sequence. So the smaller panels is something which is missing. That's why we always have this both in the genetics labs, because sometimes a clinician can ask, I pretty much know what is the requirement and all. I know only one gene is causing for that, 2 genes is causing for that. Then in this case, we can pretty much adopt NGS. If this reasons are largely unknown, we want to look at the complete chromosomes to understand what's gone wrong. Then microarray is the most economical option and higher resolution.
Since there's, obviously there's a lot of data that could be coming out of the microarray report or whatever it is. Do you have kind of tools that help us bring the data closer to the possible clinical picture? Connect both of them, because it's not a very simple interpretation. Do you have tools to connect both of them? Like maybe some algorithm.
Yeah, so when we develop these suites, when even for NGS also we have a solution, right? NGS or microarray when you're giving, when you develop the software is then back on a lot of database connectivity. So what are the current trend updated databases will be already the differences algorithms based on existing databases. So for even for NGS also, not even microarray. Based on that only your results. It's not like as such, it's just only linked with what you have designed. And even the designs is also based on the databases. Just not something we designed it and we're asking to put it in use. So the designs of these chips also based on the databases, and also this databases information will be the background and algorithms of the analysis pipelines. That way, this will be having much correlation. So NIPT is called a non-invasive prenatal testing. So I list is it out with an example. So from decades, like even from 1980s to 90s and all, even to early 2000s. So when a woman is carried like in the 3rd month or 2nd month, so first trimester you can take it. If you visit a gynae, regularly you must be hearing double marker tests, triple marker, quadruple tests and all, which is a proteomic based, analytical based markers. So this will be tested, this double marker, triple marker, quadruples will be tested is to see whether the fetus has any anomalies. Even the scan, the NT scan will be doing on the 3rd month. These are all the tools which is basically to understand is there any anomalies associated with this fetus, growing fetus. So these are the proteomic ways. One is the scanning, the NT scan will do. So another one is doing the double marker, triple markers. So NIPT is genetic marker. So non-invasive, why you call it non-invasive is, we don't need to do is any invasive like immunosynthesis. So you simply draw the blood from the mother, it's venous blood, it's an EDTA tube, and separate the plasma out from that. So in your plasma, you will have the fetal DNA. In the 3rd week or 2nd, we'll get the fetal DNA to the plasma. So we'll extract the plasma out and we'll screen that fetal DNA, whether this DNA has any genetic aberrations. Is there any genetic mutations is there in this DNA? So that you screen in the level so that we have more confidence on genetic level itself. So is there any abnormality? If there is an abnormality reported, NIPT is positive, it means that this baby may have a Turner syndrome. That also we can say. They have the Turner syndrome, Edwards syndrome and all. That can be given as a positive. This baby probably can develop. So his risk score is high for the Turner syndrome. So immediately we can do the confirmatory on this. So this will confirm, yes, this fetus has trisomy like that. So then we can able to report the clinician. Then the clinician will report back and give the counseling to the patient that. So there's a fetus. We are expecting largely an anomaly with this fetus, whether you would like to continue or would like to take it. So it's a non-invasive. So this is an advance to the double marker, triple marker. But NIPT is on NGS, next generation sequencing technology. This is a micro. 2 are different technologies, but almost hand in hand. They augment each other. There was some questions.
So following up from what she had asked, so you would say that you will extract repeatedly, like what would be the and then you will have standard protocols for that, right? What would be the percentage of chance that you might pick something from material?
It is. So when we are reporting NIPT, we something called report as a fetal fraction. So maternal cell contamination is there. So if the fetal fractions are not up to the mark given in algorithm is what we definitely report it. So what is the fetal fractions in it? The percentage should be there.
So then you recommend a retest?
Yes, if the fetal fractions are very less, what will happen is not every time the fetal DNA will be put in the given samples. It could be this could be an early pregnancy also. Because of the gestational period of like few weeks, we may not have a good fetal fraction. In that case, we can have the repeat sample collection. That will be an extra. It's not a produce, just a venous milk. It won't be paid.
Okay, so one last question. We just wanted to know, like in the primary and secondary setup. So what is your experience? I mean, like for tertiary, I believe we can afford kind of technology. Service. But what is your experience with primary and secondary setup?
So all these technologies right now, what the trend we are seeing, because we are in this market from 5 to 6 years. So what we have seen is earlier there is metropolitan cities like tier 1 cities like Bangalore or Delhi or Bombay used to have it. Now it went all the tier 2 cities. So we have seen we have these technologies in the like Coimbatore, Tirchi, and kind of even let's say we are seeing a lot of traction from even from Mysore kind of. So this is entered into this tier 2. Only the punch point here is there's only the cost. It's nothing else. Only the cost is coming, every rate is down. Because these are the same test 5 years back costing 70 to 80,000 per test. Now it's become to like 10,000. So that's why it entered into the tier 2 cities. Maybe after 4 or 5 years, there is a pretty much possibility it can even go to the smaller towns. And we do have the queries from some kind of towns from Gulbarga also. Belgaum, Gulbarga, and Dharwad also we have queries. So people are investing in that. It will reach tier 3 something is futuristic for us, but as of now it's tier 2 is there. So wherever this opportunity is not there, so the reason since it is required only the DNA, it doesn't require any culture, no transport content, nothing. If the tertiary, I mean the smaller cities and all, if at least you have the basic setups for extraction of DNA, storing the DNA, -4, -20, -80 degrees temperature. At least they can extract the DNA and they can transport this DNA. Fortunately, we have the logistics better now. Anywhere in India, 24 hours, we can able to ship anything. So in that case, they can have the basic setup of extraction. That's what even the bigger teams are doing, like our service providers, people are buying our technologies. So they are opening a small centers, small hospitals in the towns. This is a collection center. They'll collect and they pre-process it and they safely pack it and send it to the cities. So they are analyzing it. So the reach is good. Investment wise, it is still not into the tier 3. It's limited to as of now is the tier 2, but it's expanded. Any questions, sir? Sorry. So if you anybody has any questions probably around this technical or something, so Dr. Atla is there, so probably can ask her. So we can able to address any kind of question even later of the presentation at any point.
Just one last question.
Yes,
What would be the cost for basic model?
So this one cost you close to 1.5 crores setup. So that include your qualification, validation, and everything. So all PA MBBS doctors.
Yeah.
These are the latest advanced technologies. We have to bring these technologies to the practice clinical, which will be much more useful for the diagnosis and treatment of the patient.
Hopefully in future it becomes like a CT or MRI.
It will. That's where it's stepping in actually. So we have replaced the large karyotyping requirements with these 5 years. This has been regularly used and paid by the insurance companies in abroad. Since we are cost-effective people, of course, I cannot pay the 50,000 test and all. So we are cost sensitive people, no insurance is covered, but growing market. But definite requirement.
Questions and probably I thank Tom Fisher and Raju and his team for being here and sharing the knowledge with us. The primary focus of this workshop, as you know, it was on karyotyping and FISH, but this aspect, I wanted you all to know that you have something more than that also. That's why we have kept a short session on this. It requires a whole day session to understand how the data analysis is done. So, but still you have some ideas, and when you start using these technologies, you will not be totally, caught off guard that you don't know anything about. Something, some idea is always important for you. Thank you, Rashid, for
Thank you. Sorry.
Good afternoon, everybody. Now this session is back again to the karyotyping, which Dr. Vijay Madam had spoken. So this is going to be a continuation of that one, more of technical aspects. But then I'll touch upon few karyotyping principles behind the technical aspects also. And then I will move on with the video presentations, probably because we have very less time of 1.5 days to show you completely both cytogenetics and FISH. So if we go with this video, then you'll understand better when we're doing the analysis in the workshop. So.
Okay. Next. Yeah. Next one. Just check. ಇಲ್ಲ secondary. ಮಾಡ್ತೇವೆ. Little next ಆಯ್ತು. ಎಷ್ಟಲ್ಲಿ ನೆಕ್ಸ್ಟ್ ಇಲ್ಲಿದೆ ಫಸ್ಟ್
So I'll be talking about the complete genetic diagnostic technique and their application in medical practices. So going with genetic testing, it's become an integral part. In the inauguration itself, we have spoken about it, where this cytogenetics has become the integral part of diagnostics in almost all the hospitals. So first of all, we'll have to go with the applications where all it is applicable, this genetics part. So it is applicable in the constitutional genetics, that is the clinical practice. It's in prenatal diagnosis, which is very, very important because the genetical abnormalities are not curable. Okay. you must be knowing if an individual is born abnormal with genetical disorders, it is not curable. So the best way is prevention. Prevention is better than cure. And once the child is born, the curative cost and the maintenance and the management is very, very difficult. So prevention is best. So prenatal diagnosis is very, very important in genetical testing. Next comes postnatal, then the pediatrics, reproductive disorder, endocrinology, and psychiatry as well. Generally for the schizophrenia and Fragile X and all those things, we get cases from psychiatry as well. Other applications apart from constitutional genetics is oncology, which we are not talking much today. And next is in the public health program, where it is very important in the premarital screening, especially in the couple with the risk of inherited diseases and newborn screening. Next. Yeah. Now this is a comprehensive slide of complete genetic testing. So here you can see the genetics testing can be divided into two, that is molecular diagnostics and cytogenetics. So molecular diagnostics is more of the DNA assays and then the analysis. It is more of the mutation test, and it is either single gene mutation or multiple gene mutations, and it is all at gene level. Whereas cytogenetics is at chromosomal level, and generally cytogenetics is involves FISH, karyotyping, and microarray, which the next Mr. Raju spoke about it. So FISH, Dr. Lakshman will be speaking, and karyotyping, I will be talking. Now, so where are the constitutional genetics? Where are the cytogenetics may be applicable? It is in premarital screening or premarital counseling also we have. There, a couple who is going to get married, if they already know that there is a genetic disorder running in the family, they can do prenatal, they can do premarital genetic testing to just check if they have any kind of genetic disorders, which they can prevent it in future after the marriage. Next comes pre-conceptual counseling, especially in IVF centers. If there is, they get to know that there is some genetic disorders running in the family, or there is some difficulty in conception because of which they are not having babies, then pre-conceptional genetic testing is required. Next comes prenatal diagnosis. These three are preventive measures. All these three are preventive testing. So prenatal testing, again, it is before the birth. During the antenatal testing, we have certain genetic tests done. So prenatal testing can be done. Next comes newborn screening. All these are very much important in genetic testing. So basically what this, how it is important is, it can prevent, this is same application for this as well, not only from cancer. It can have a prevention method, prognosis and diagnostic, and also therapeutic. Now, the causes for genetic abnormalities or malformation. Apart from genetic disorders, which is inherited, it can also be environmental, like, multifactorial, apart from the chromosomes, apart from the chromosome and the mutation, it can also have multifactorial. It can be environment, drug-induced or infections, or any exposure to radiations or chemicals. So coming to various genetic testing, first, I would tell about the conventional cytogenetics and the molecular cytogenetics, which involves FISH, M-FISH, that is multiple color FISH, and M-banding, and microarray also. But here we will be using predominantly the regular FISH, for analyzing the chromosome number and the structural abnormality, the cryptic abnormalities, especially the one which are really very subtle chromosomal variations or breakages or translocations. So coming to molecular, it involves the other tests like PCR, MLPA, sequencing, and NGS. So again, these are all chromosomal level and these are gene level. So coming to just understand the levels of genetic testing, here you can see this is the chromosome and the karyotyping, where you will be seeing the number of the chromosome or the aberrant chromosome, where Vijay Madam had explained to you all about the structural abnormalities and any change in the number of chromosomes. Next comes the cryptic region, which we detect by the FISH techniques. Here you can see this would be a whole chromosome. FISH would be the small segment on the chromosome. And next comes chromosomal microarray, which will be much more in detail. In this region, we will be going much more in details of the chromosome. And then, of course, at DNA level, we'll be going to the base pairs. Now, at karyotyping, here you can see the chromosome numbers, as well as the structural abnormalities. Any change in the number or structure of the chromosome will cause the abnormality. And FISH, here you can see it is basically the fluorochrome or the signals on the FISH which you can detect. So generally, we all have diploid sets. Generally, all the chromosomes, they have to have two numbers. So here, just to show an example, here we can see two signals of green and two signals of red. That means it is normal. Here, if you see two signals, green and 3, that means this is Down syndrome. It is showing trisomy 21. This is the difference between the karyotyping and FISH. These are all more of signal patterns in FISH. So I'm not going more in detail because Dr. Vijay Madam has already spoken about the chromosomes, and practically I will be teaching you all in the workshop. So these are the chromosomes, just to have an idea how the chromosome looks like and the banding pattern. These are the acrocentric chromosomes. Now, this also Madam has told, I'm just going fast a little bit because I want to show you all more of the technical aspects. Now, who should do this cytogenetic test? So there are certain cytogenetics indications. It can be prenatal, postnatal, or the adult or the neonatal. So generally, any couple with repeated miscarriages or stillborn babies, they can undergo karyotyping. And then any abnormal pregnancy which has IUG or GR or any congenital abnormalities of the fetuses, they can be recommended for karyotyping. Then any individual with congenital heart defects like BSD, AVSD, double outlet right ventricle, etc. Any congenital defects can be recommended for karyotyping or genetic test. And skeletal abnormalities, ambiguous genitalia, delayed puberty, or the secondary development is delayed. These are another indication. Short stature, primary and secondary amenorrhea, oligospermia and oospermia. This is also recommended because you can have chromosomal, Y chromosome deletion. Then chromosome breakage syndrome, like Fanconi's anemia, ataxia, Bloom syndrome, etc. And suspected family history of chromosomal abnormality. Suppose an individual is having repeated abortions or a child with dysmorphic features, we can always recommend for karyotyping, where you can suspect that there could be some chromosomal abnormality. Now that is postnatal. I'm going to specific more about the prenatal. I think your workshop is more about prenatal. So the indication for prenatal is if a couple has a previous child with Down syndrome. So any couple who has a previous Down syndrome definitely will be recommended for karyotyping. And in advanced maternal age, if a woman is above 35 years, karyotyping is recommended. The reason why is above 35 years, the chances of non-disjunction of the chromosome is more, because the chromosome, you must be knowing about the meiosis. Okay, so in the meiosis, during the anaphase, there could be a lag of the chromosome moving to the polar regions. Because of that, there could be non-disjunction. So it is always recommended for advanced maternal age. Triple test positive. Apart from this, now quadruple is also come. If quadruple is positive also. And here one more is missing, NIPT is also missing, non-invasive prenatal diagnosis. If that is possible, a positive, definitely a karyotyping is recommended as a confirmatory test. Then family history of Duchenne muscular dystrophy and any other single gene defects or sickle cell or thalassemia, definitely this is more of molecular, not karyotyping, but prenatal testing is recommended. And inborn errors of metabolism and carrier of the balanced translocation. If the parent is a carrier of balanced translocation, generally what happens, if the couple is having repeated abortion, they would have done the karyotyping, right? So if any one of the couple is a carrier of a balanced translocation, the fetus has to be detected. The reason why is the child or the fetus can have an unbalanced translocation. During the meiotic division, there could be unbalanced translocation, which the segregation of the chromosome can take place. Because of the unbalanced segregation, the fetus can be abnormal. And then the fetus, when they go for antenatal checkup, definitely ultrasound is done. And if there is any fetal anomaly, which is detected. So let's see what are those ultrasound anomaly. It's not that all the anomalies has to undergo karyotyping. There are certain indications for that. So these are some of the abnormalities which is found in the ultrasound, like increased nuclear thickness. This is the ultrasound in the first trimester which is done. If there is increased nuclear thickness, definitely a karyotyping has to be done. Congenital heart defect, skeletal abnormality, cystic hygroma and fetal hydrops. This is a very good marker to suspect Turner syndrome. Then gastrointestinal and renal abnormality and CNS abnormality. If any of this finding is there in ultrasound, definitely a karyotyping is recommended. So this is just to show, I have got my heading over here, it's not visible. These are some of the abnormalities found in pediatric clinics, like Down syndrome, Patau syndrome, Edward syndrome is trisomy 18. So some of these are the example. And these are adolescents, generally like primary amenorrhea or even some of them secondary amenorrhea. You can have either Turner syndrome or the male can have a Klinefelter syndrome where XXY you can find. Yeah, this is the BOH case with a bad obstetric history. The reason why I put this is a very nice pedigree. You can see there are 2 generation pedigree where repeated abortions are there. Only one normal child is there, but there are consecutive repeated pregnancy losses. And if you see here also there is a history of pregnancy losses. So when this couple came, we did a karyotype for a couple. And we found that there was a translocation on with the mother of 14 and 21. Here you can see there is 14 and 21, which is translocated onto the 14, which is called as Robertsonian translocation. And this is another a pedigree chart, which they had come for prenatal diagnosis. Again, we have done, and the father was the carrier, and this child was an unbalanced translocation with this extra part here, and the father was a balanced translocation. Okay, and unfortunately, this fetus was balanced like the father. And this is a case of prenatal diagnosis, where here you can see a consanguineously married couple. The previous child with Down syndrome. So we have coffee. So here you can see this couple. They were consanguineously married. They had one child which was normal, another child with a Down syndrome. So when they had come for prenatal diagnosis, we found that there was a cardiac defect. And when we did the karyotyping, you can see that there is a deletion, interstitial deletion of this band on chromosome number 8. So when we repeated the scan after at 22 weeks, this child also had the oligohydramnios and also cleft lip and palate. And then they decided, because already they had an abnormal child, they decided to okay. And if you see the autopsy, the child had cleft lip and palate and oligohydramnios. So now, what are the samples required for prenatal diagnosis? As madam has already told you all, we need certain samples for each test. Here for prenatal, generally we require chorionic villi sample, amniotic fluid sample, or the cord blood cultures, which we put. For postnatal, it is generally pelvic blood. I'm not going through the cancer. These are the things required for the prenatal and the postnatal test. So for karyotyping, generally we require heparin. It is not the EDTA. EDTA is only for required for molecular testing, but not for karyotyping. Now, we generally require 1.5 to 3 ml of venous blood in the heparin vacuola. Okay, it is not the EDTA, because why we stress upon this many a times, if the clinicians they don't know, they send it, they send it an EDTA sample for karyotyping. We cannot do the test on EDTA sample. Now these are just to show the prenatal sample. Here you can see chorionic villi, generally it is done between 10 to 12 weeks. So the sample looks like this when it comes to us. We have to tease out and clear the chorionic villi, and the villi we have to observe under the microscope, and this is how the villi looks like. And between 16 to 20 weeks, amniotic fluid can be taken, and the sample has to be sent. All these samples has to be sent in a sterile condition, because all the karyotypes are in an aseptic condition. It has to be sterile because we have to culture it. And when it grows, the amniocyte looks like this in the plasma. And then now the cord blood is generally like the regular cord, the blood sample. But of course, for cord blood, again, maternal cell contamination test has to be done. Now, coming to the technical workflow for the peripheral blood, most of the culturing technique is almost same for all the indications. So the material required is centrifuge tube, pipettes, flasks, and sterile tips. And the equipment is biosafety cabinet, carbon dioxide incubator. Apart from that, you will need centrifuge and vortex as well. And the reagents what you require is RPMI media, fetal bovine serum, and PHA, hypotonic solution, and fixative, and the calcium. Now coming to the principle of these reagents, PHA generally enhances the growth of the cell. And why I'm telling this one, when we are doing the workshop, you should understand why we are using these chemicals, right? So the media is generally a cell, it stimulates the cell division in the cell culture. And the cosmet of colchicine, here the cell division, we have to put the culture for 48 hours or 72 hours minimum. Okay. So when it goes under cell cycle, we have to arrest it at metaphase stage, because all the karyotyping is done at metaphase stage. So to arrest it at metaphase stage, we use this calcimet. Okay. Now ethidium bromide is not used anymore, but then initially we used to use it. But ethidium bromide is used even now to get longer chromosome for a higher resolution. Okay. So generally it promotes chromosomal elongation. And KCl is the hypotonic solution. It allows the cells to swell up. Okay, when it swell up, it's like the cell, like a balloon. Once you have a balloon with the water filled in and put it down, it breaks, right? Similarly, the cell swells up. You put it in a hypotonic solution, it swells up, and it allows the chromosomes to come out of the membrane. Then, of course, James's solution is used for stain. Now, the reagents and chemicals required, I've already told you. The hypotonic solution, media, all these things. Yeah, of course, for slide banding, you need the buffers, normal saline, and the trypsin for the banding. Now, coming to the culture, generally these are the things required. It's just a repetition, but this I can just to show the image over here, because we cannot show much over there. If it is here, all of you can view. You generally need the media, the sample. For the blood sample, we need a centrifuge tube. If it is amniotic fluid or a CVS, we need a flask. Okay, of course, the tips and the micropipettes. This is the PHA. Now, just to show Yeah. Just to show, before we start any cultures, the area of the culture has to be thoroughly cleaned with 70% alcohol or ethanol, completely clean, because and the airflow has to be continuously running so that it will be sterile over there. If possible, every 1 month fumigation has to be done so that the laboratory is sterile. Yeah. Here you can see around 8 ml of sterile RPMI media, which is a media for growth of the cell culture. You can see here he's adding the media into the tube. Around 8 ml or 5 to 8 ml of 8 ml is enough for the culture. So this is the PHA, around 200 to 300 microliter of pH is added into the media here. pH is the stimulating agent. He is adding 300 microliter. Next. Now you get the media with complete media with the serum. Otherwise, generally what we do is we use 15% fetal bovine serum. But nowadays, like it's become very commercial that we get ready to use media. So no need to use serum. And you get even with PHA also. If you don't have this media, if you're separately preparing RPMI media, then along with the media you have to add PHA and the fetal bovine serum. Okay. Now, once the blood is dropped into it, after the pH is added, now the sample is added into the tube. Then gently mixed. Once the sample is added, it is gently mixed. And once it is mixed, samples are also labeled. If you have multiple samples, it's always better to label the culture tube or the task before you start the experiment. Now, this was the demonstration, so we have used only one sample. But if you have multiple samples, it is always better to label it before you start the experiment. Now, once the media, the PHA, the serum and the sample is added, it is gently mixed and then kept in the incubator. Generally, 48 hours to 72 hours culture is kept. Minimum at least 48 hours has to be kept. Except for cancer sample, overnight or 24 hours can be done. But generally for the postnatal or the regular blood sample, it is 48 hours. And we prefer for the pediatric sample, generally we prefer 48 hours cultures. Okay, because the way they take the blood, especially in NICU, when we get sample from NICU, we prefer 48 hours cultures. Okay, so it is kept at 72 or degree, 70 hours. But generally, we start resting the culture at 68 hours, and it is kept at 37 degrees centigrade, and it should be in a carbon dioxide incubator. And another thing is, like alternate days, it is always recommended to take out the tube, loosen the cap, and then keep it back, gently mix it and keep it back. The reason why the carbon dioxide, which is released out of the culture during when the culture is growing, if you release the carbon dioxide, which is there in the tube, it enhances the growth much more. Yeah.
So, videos.
Yeah. This once after, this is actually day 4. That means you are already, that is day 1, the culture. This is day 4. Day 4 is already you're into 70 hours. Day 1 is counted the first day also. When it is 37 degrees centigrade, I mean, when it is 67, when it is 68.5 hours, the consummate is added. Okay. Once the consummate is added, previous slide only.
Previous one out.
Previous. Next. Previous.
Select
Previous.
previous.
Yeah. See, after 67.5 hours of incubation, you should. Here, what happens after 67.5 hours? You add 50 microliters of consummate. What is consummate? I've already told you. It is the reagent which arrests at metaphase stage, because we need the chromosome at metaphase stage. So once it is 67.5 hours, we add consummate around 10 microliters, and then further incubated for 60 minutes. After the consummate is added, again it should be incubated, because the consummate action has to take place. So here, once 60 minutes is over, the incubation with the consummate is over. It is set for centrifuging at 1500 RPM. Between 5 to 6 minutes also you can output.
Incubation insert incubator, take the sample, mix it properly and centrifuge it.
So once the centrifuging is over, once you centrifuge, the supernatant will be the separate clear supernatant. Then you can discard the supernatant and just have the red pellet down, which is there. Okay. So you can see there, this clear thing, it can be discarded. Only this pellet has to be kept for us. And you can see a little bit of buffy coat over here. So till here we can remove. We shouldn't fully remove. We need to have at least till this follicle region. Yeah.
Discard the supernet.
Supernatant is discarded.
Only the supernet, not the deposit. Okay.
And this is rotate testing after the supernatant.
Be careful while discarding.
It can be rotate the pellet over there. That will gently rotate it.
Now,
Yeah, this is rotation. Once you rotate, you have to add the KCl around 8 ml. KCl is the hypotonic solution. This is actually rotation. After that, the KCl is added.
acetic acid.
Okay, that step is missing over there. So once the vortexing is done, the pellet is there, the hypotonic solution added, that is the KCl is added, and it has to be incubated. Generally, the incubation of the hypotonic solution varies from laboratory to laboratory and also based on the temperature of the laboratory. If the laboratory is really very warm, only 10 minutes of incubation is enough. But if it is colder, the colder laboratories or the region, they can increase even up to 20, sometimes even up to 30 minutes. But generally, the temperature of the hypotonic solution varies from laboratory to laboratory. So once the hypotonic solution is done, after the incubation, again you spin it, remove the supernatant, and then add the fixative And this first fixative has to be cold and freshly prepared because the ratio of the acetic acid and methanol should not be varying. So always be prepared fresh. The first the fixative has to be really fresh and it has to be cold. And the first fixative has to be really very slow because if you rush and put the fixative, you will start getting kind of debris like of a pellet. So it has to be dropwise and very gentle. See, it's all very slow and gentle. And then after the similar kind of fixative changes, at least minimum of fixative changes are taken.
Put some more fix for that.
Slide replacing Nimisha.
So that the
After 2 fixative changes, the slide preparation is done. So 2 fixative washes you have to give. Once you finish the fixative washes, to clear the RBCs, all the RBCs which are there, because you need only the lymphocytes, right? The lymphocytes, right? So you have to remove the RBCs completely. So you have to give 2 fixative changes. Once 2 fixative changes are done, then the cell pellet is adjusted and then the sliding casting the slide takes place. This is the fixative, the similar to. After that, you have to do the slide preparation. Now here the slide preparation is here.
See the turbidity of that. If it is too much turbidity, put some more fix for that. So that the concentration will be correct.
Here you shouldn't dilute too much also because you need to have sufficient pellet in the slide when you're preparing. Just till the corner of the tube you can prepare the pellet, just till this corner. If you feel the pellet is too much, then you can dilute it much more. Next slide.
See the turbidity of that. If it is too much turbidity, put some more fix for that. So that the concentration will be correct. Feels that this is fine. Or little more.
Next. Now the slides are prepared here. Slowly the slides are washed with fixative again because you want the slides for the metaphases to spread evenly. So you have to put few fixative drops onto the slide. And then the pellet is dropped. From here, generally what happens here, this technique also, laboratory to laboratory varies. Generally we hold the slide and we drop it at a height, okay? So that the cells burst out of the membrane and spreads onto the slide evenly. So again, this technique varies from laboratory to laboratory. Some of the slides are that standardized, just they just drop it like this also. But we are all practiced to drop it from a height. Okay, so this is how it is prepared, the slide. And while the slides are dropped, it has to be heated on the hot plate. Next slide would be there. It has to be heated on the hot plate and then kept for aging at 60 degrees only.
Okay, like this. And take one drop of your
yeah. So once the slide is prepared, you can just observe in the microscope. The first one is called as the test slide, where we test for the index of the chromosome. Index means like number of chromosome or the metaphase. I mean the chromosome spreading and the number of metaphase. We have to have sufficient. You shouldn't make too dilute. There you have to, because karyotyping is very time consuming. If you have too dilute, you waste a lot of time during the scanning of the during the analysis. So it has to be a little bit, you shouldn't be too dense also, because if there is overcrowding, the metaphase won't spread out well. If it is too dilute, you will waste time in scanning the slide. So you have to adjust in the first slide. First slide is always called the test slide, where you check for the index as well as the spreading of the chromosome. So we have to check that one in the microscope, and then you keep it on the hot plate. This is the hot plate. Generally, you keep it for baking there for sometimes during when you're preparing the slide. Once that is done, it is kept in the hot air oven at 60 degrees centigrade, and it is aged for overnight. So once you age, you have crisp chromosome for banding the next day. So you will for those risk chromosomes, the slide has to be baked overnight. Yeah, this here you can see the slide over here. This is the slide over where the slide can be dried here. It's just doing that because down part also has to dry. And it's just we can keep it on top.
See at the microscope.
So that was one which is already prepared. He is observing under the microscope just to see if there is sufficient index and the spreading of the chromosome. If this low. Next one. Now next after the slide is the bake, you have to go for banding. Here you can see there is 4 coupling jars. Here the first jar is going to have the trypsin working solution, that is 50 ml of normal saline and 5 mg of trypsin powder, or you get even the liquid trypsin. And the coupling jar, which is the next one, which has 50 ml of normal saline, and the third coupling jar has the buffers. Okay, so you have the combination of buffers, and the fourth one is the normal saline. This one also has the gypsum stain in it. So we go through these steps for the banding. Here you can see which is arranged one after the other. So it goes, and the trypsin action has to be timed also. So again, the trypsin action varies from laboratory to laboratory. So here, the slides are incubated, which I've already told you, at 65 degrees, and then it is trypsin action takes place. This, I told you, this timing varies from laboratory to laboratory. Generally, it is around 20 to 30 seconds or half a minute also. Then brief mixing, rinsing in normal saline, and then the gypsum stain for around 10 minutes, and then again rinsing in distilled water. And then once the slide is banded, you shouldn't mount it immediately because you can't store for a long time if there is moisture content. So it has to be thoroughly dried before you add. Mount the slide with the TPX. So once it is dry, it can be mounted with using DPX solution. Next. So next, once the slides are banded, it is ready for analysis. Here you can see this is the microscope here, which we have. This is the microscope which we use for analysis. And here you can see the stage over here. We mount the slide here, and then we have a camera. And this camera is fixed to the monitor over here, where we have the software also, which does the karyotyping. Okay.
Which is used for karyotyping. Here you have the microscope, which has got the lenses here. We have 10x, 70x, 100x, and a few other filters, which is for fish. We have the DAPI filters here and all the fluorescent filters here. So what we do, once the harvest is over and the slides are prepared, the slides are taken and kept here. This is the stage to load the slides. So once the slides are loaded here, we analyze the slides by moving this stage here. This is the stage which moves up and down, x-axis and y-axis. This is how we move the slide. So once the slide is mounted, and then we choose the right chromosomes in the metaphase, and it gets captured here with this camera here. So once it is captured, this image is transferred to the system where all the softwares are loaded, and we do the analysis. So I'll move on to show the analysis, how it is done. We have got the slides, which is banded, casted, and which is ready for analysis. Once the slides are ready, it is mounted onto the slide here. This is the stage for mounting the slide. So once it is mounted, we go to 10x. This is the 10x.
First, obvious scanning is done at 10x.
Here, the microscope is switched on, and once the light comes, you can increase and decrease in the microscope. So here, what we do is we scan the slide for various to find out various metaphases, which is of good morphology and good in structure and number as well. So what
you have to pick up the slide, the metaphases which has good morphology also, not randomly cannot capture.
once we find good metaphase, I found it at a good one at 10x. So we heat this oil emulsion. This is the oil which is used to view it under 100x. So from here it is moved on to 100x. So this is 100x objective. So under 100x objective, it is more clear.
The analysis is generally done in 100x. And
Once we focus it, this is how the chromosome looks like. We have got a slice.
So once you view on 100x, this is how the chromosome looks like. I've just put few metaphases with the GTG banded, that is banded. So this is the chorionic villi sample. This is how generally the morphology of the chromosome looks like in chorionic villi sampling, whereas amniotic fluid and the blood sample or the cord blood looks much more better. So generally, this is how the chorionic villi sample looks like. And always for the structural abnormalities of the chromosome, generally we don't request to do the chorionic villi sampling because we tend to miss out the structural abnormalities in this kind of morphology. So always amniocentesis is recommended. Next one. So in 10x, which I told where we scan the metaphases, this is how the chromosome looks like. Here you can see, if we see around 5 metaphases in one field, we say that it is good index. And also you see very less of overlapping. You shouldn't have the crowded metaphases like this. You should have very minimal overlapping. So you get banding also, the quality of the banding also should be checked before going ahead for analysis. So this is how you can see the chromosome looks like in high power. Next one. So just to show the final karyotyping after the software, here we cannot show the demo of the software, but we'll be showing in the workshop there. So this is how the final chromosome looks like. This is the normal XX and this is the normal XY. So the banding, based on the banding, the position of the centromere and the size of the chromosome, the chromosomes are classified, which Dr. Vijay Madam had explained to you all in the morning. So that's it with, yeah. So again, this is the ISCN, which we do use for the analysis and for the reporting guidelines. Actually, this is the old version. This is actually 2020, which we have got now. So this is the guidelines which we follow for reporting and for the analysis purpose. So that's the end of my session. If any questions, then let me know. And again, for the technical part, we can see more in detail in the workshop. With this, you have any doubts, you can let me know. Any questions? No questions? Yeah. I request Dr. Lakshman from Meta Systems to do the talk on application of FISH in clinical diagnosis.
As many things. Plus.
Sit. Oh. Oh.
Let me just next. Let's just point it again. That. So, good afternoon, everyone. I'm Dr. Lakshman. I represent Meta Systems India. So, topic for today is applications of fish in clinical diagnosis. Obviously, I will be talking more about fish. I'll try to finish it a bit faster also because all of you are lunchtime here. Let me start. So, we are a German-based company. Since 1986, we have been into metaphase scanning systems, automated metaphase scanning systems. So these are our founder, Dr. Thomas Losch. He was the one who developed the first algorithm for scanning metaphases on slide and dissecting. So in 1983, it was a part of his thesis, PhD thesis at University of Heidelberg. So we had the first automatic slide scanner ever. And this is our chronology for the timeline of ours. We had the first metaphor in 1985. That is the non-commercial installation at the University of Heidelberg. Then Meta Systems company was founded in 1986, followed by commercial installation. First commercial installation in 1987. Okay. Iteros software. Iteros stands for Interactive Karyotyping System. It's a software for karyotyping. Software karyotyping. First developed in 1991. Subsequently, we developed the FISH imaging software, which is known as ISIS, In Situ Imaging System. Okay. So ISIS stands for In Situ Imaging System. We developed in 1994. Then we came up with higher end probes in 1998. MFISH and M-band probes. MFISH for multicolor fish, M-band for multicolor banding probes. Okay, so these are all higher end probes that we developed first, and subsequently we came down to locus specific probes in 2009. Okay, so this is our probes that Dr. Elsie showed about. So she has the patents for M-band. Okay. And 2004, our operations started in Asia. We have a head office in Hong Kong, and from 2012, we have our operations in India. So today, we take care of all these metaphors and the softwares for karyotyping and fish. Okay, so we have a big market in India, and most of the well-established labs will come across our systems for automated scanning and also our softwares for karyotyping and fish. Okay. So we have more than 6,000 workstations worldwide. So this, we are not only concentrated on metaphase scanning as such. We have diversified into various disciplines of biology. So the clinical diagnostics, of course, is our forte with cytogenetics, hematology, and clinical oncology. We also have solutions for pathology, digital pathology, where we have modules known as V-slide, which stands for virtual slide viewing, where you can digitize pathology slides and analyze them at your convenience. Okay. Then we have solutions for toxicology and radiation biology. So toxicology, it's all about genetic toxicology, of course, with respect to your assessing genetic damage by assays like the micronucleus or the comet. Okay. So we have softwares for all of them. We have also ventured into microbiology, where we have the automated scanning of gram stain slides and also microbacterial scanning. So, and we also have a technology similar to FISH in microbiology known as DMI, direct multiplex imaging. Okay, where you can view or analyze bacteremia samples, and you can analyze within half an hour in primary blood cultures around 15 strains, bacterial strains. Okay, so that is one thing here in microbiology. Then forensics, we are into sperm counting and scanning softwares. Okay, so but all these require the automated scanning system, scanning and analysis platform known as Metaphor. Okay, So I won't go to the cell cycle because it's been continuous lectures for you, starting from the basics of cytogenetics. So this slide I just wanted to show that we are going to deal with metaphase and interface. So throughout the workshop, you'll be coming across these terms, mostly the interface FISH and metaphase for karyotyping. Next. So this is how interface nuclei will look under a microscope on the left, to the left. Metaphase will look left to this microscope. So all this has been discussed. We have the chromosomes, we have P and short, P and Q arms, P for the short arm, P for the long arm. Next. This is the workflow that Madam was discussing some time back. You have the venous blood, you have the phytoheme or glutenin, which is a mitogen which stimulates your T lymphocytes in culture. Then you need to culture it for 3 days at polycystin, which will arrest your tubulin polymerization. Okay, arrest the cells in metaphase, and then you will fix the cells using fixative known as carbonized fixative, which is methanol and glacial acetic acid in the ratio 3:1. So for conventional cytogenetics and also for FISH, you will be using this carbonized fixative. Okay, so most of the genetics labs you will come across these terms. Then you need to spread the cells by dropping them onto these slides. The fixative will help in scattering of the nucleus, okay, or the metaphase squares. Then you digest it with trypsin, which will digest your histones, and you stain it with GMSA so that you get alternate dark and light bands. Alternate bands, dark bands refer to the AT rich regions, light bands to the GC rich regions. So this is the karyotyping done by our software. So this is a case of 46X female. Okay, so three things as we discussed on the morning. One is the size, the centromere, and the banding patterns. So based on the size and the centromere, the chromosomes are grouped from group A to G. Okay, so next. These are different groupings. So 1 to 3 is group A, 4, 5, 6 to 12, and X is the group C. Okay, metacentric, also submetacentric chromosomes. Group C, heterocentric, E is again metacentric, group F is small metacentric. After this is 19 and 20 chromosome, followed by 21 and 22, group G, that is heterocentric, and the Y chromosome. So all this is the basic grouping. This has also been discussed. So what is the notation or what is the designation? At this juncture, I would like to mention that we have with us the DNN-based automatic karyotyping software. It is the fastest in the market available. Okay, DNN stands for an algorithm known as deep neural network, which is a type of artificial intelligence-based algorithm. Okay, so in this, in cytogenetics, it's more about skill. The machine will do a bit for you, but with AI-based softwares, with just 2 corrections, you can get 99% of the karyotypes right in case of blood and 95% in case of bone marrow, which is a huge thing. Okay, because otherwise the skill of the cytogenetist is everything inside in a report. Okay, so based on the bands, the cytogenetist has to take a decision. But with this, the arrangement of the karyogram will be 99% accurate. Okay. And we have the error rate in the case of lymphocytes is less than 1. That is a rarity. And for bone marrow, again, bone marrow is very tough to karyotype. So it is less than 10. Okay. So it is the best in the market as of now. And we have it for different banding types, G banding, R banding, and P banding as well. So coming to the abnormalities, we have. I'll be concentrating mostly on the abnormalities that we target to FISH. Okay, FISH stands for fluorescence in situ hybridization. So these are the abnormalities that are usually targeted. They include deletions, where a small portion of the chromosome is missing, or is deleted. These are structural abnormalities. Okay. Deletions, duplications, where we have duplication of a gene. Okay. Translocations, again exchange of partners. Chromosomal rearrangements between two chromosomes. Okay. Like the case of 922. We said. Then inversions, the genetic segment gets inverted. Okay. So that is mostly with respect to intrachromosomal aberration. Okay, pertaining to a particular chromosome. Then you have the numerical abnormality. So you have the structural and the numerical. Numerical, we are going to, we'll be talking about the basic syndromes and also polysomy in chromosomes in cases of cancer. For example, leukemia, you will have polysome of 8 or next. Next. So these are all have been discussed in the morning. Just pass forward. Next. So coming to FISH, FISH stands for fluorescence in situ hybridization. So a powerful molecular cytogenetic technique used to detect chromosomal abnormalities or aberrations, highly sensitive and specific. And both types of nuclei, the metaphase chromosomes and the interface nuclei are used in FISH. So it is known as interface FISH or metaphase FISH. Okay. So you require a fluorescent labeled DNA probe for detection and analysis. So the advantage of FISH as compared to the conventional cytogenetics is the time or a short turnaround time. So conventional cytogenetics, the report takes around 8 to 12 days. Okay. Whereas in FISH, anything between 2 to 4 is set by the labs. So it is even shorter than the NGS or the microarray. But the thing is, FISH is more specific. Okay, it's going to target only specific chromosomal abnormalities, and it can be used in single cell analysis also. Okay, and analysis is also not very complex. You need to check 100 to 200 nuclei per sample. Okay. And this will give you an information for a targeted therapy or treatment. So this is a pictorial representation of what the technique is all about. So you have the double stranded DNA in the sample. You have the double stranded probe. Okay. So there are 2 steps. First step is known as denaturation, where you need to denature the target and the probe. Okay, sample DNA and the probe. Then you need to hybridize. So second step is hybridization, which is done overnight. So denaturation is done here at 75. Okay, for anything between 2 to 5 minutes. Then hybridization is always 37 degrees overnight. Okay, so this can be done in a FISH hybridizer, or if the hybridizer is not available, you can use a hot plate and the incubator, normal incubator. Okay, 37 degrees. So what are DNA probes? They are specific segments of DNA targeted to a specific region of the template sample DNA. So the smallest resolution with FISH is around 100 kbs. So most of our probes, if you find check our catalog or our website, you see that they are around 400 to 500 kbs. All these probes. Okay. So that is the resolution. Once the probe is selected, the fluorescent labeling. Nowadays, everything is done by direct labeling. Okay. So we use different techniques, chemical-based techniques to label the probe. Okay. So this is just the basic diagram of fluorescence microscope. You have a mercury source, then you have the filter tube here. Filter tube has the emission, excitation filter and the emission filter with the dichroid mirror. Okay. And here it's all going to be reflected light. So this objective here will serve as the condenser. Whereas in normal light or bright field microscopy, you have condenser here and light source from the base. Here, this is the pathway of the light, and it's going to come through a excitation filter, move through the dichroic mirror onto the sample, which is labeled for fluorophore. The fluorophore will get excited, okay, and it will emit fluorescence of a higher wavelength. It will be excited. The sample will get excited by a shorter wavelength of light, but then it will release a fluorescence of a higher wavelength, which will move through the emission filter and onto the eyepiece, or will be captured. Image will be captured by a camera. So mercury lamp is the usual source for your UV, which is going to fall on the sample. Okay. So this is the basic setup. So if coming to fluorophores, you have different probes, different fluorophores. The first three are the universally used fluorophores. U stands for aqua, okay, also known as DEAC or spectrum aqua. Green stands for FITC or spectrum green. Orange stands for TRITC, spectrum orange. So these three are normally used in most of the probes. Okay, dual color and tricolor probes. In addition to it, there are two more which are used in special probes like the multicolor FISH or multicolor band. This is the Texas red or spectrum red and near infrared, which cannot be viewed under the microscope but can be captured by the camera. Okay, so they have different absorption and emission spectra. So coming to the FISH workflow, so just need to isolate the sample, and you have to make sure that the sample isn't fixated for FISH to proceed. Okay, so sample can be anything, peripheral blood or bone marrow aspirate, and you do some processing with KCl and fixative. Okay, then you keep the cells in fixative, then you drop slides, apply your probe. So once you drop the slides, you just need it that fixative dries up, then you apply the probe, seal it with rubber cement. Okay, so that the probe does not dry up while you're taking it for denaturation. Okay, so next you use a FISH hybridization for denaturation hybridization. It makes the work simple because it has got stored programs which can be used from time to time. So the denaturation is 75 degrees for 2 minutes. The hybridization is 37 degrees overnight. And next day, you have to do the post-hybridization washes. So post-hybridization washes are done using a buffer known as SSC, saline sodium citrate. It's a combination of sodium chloride and sodium citrate. Okay, you use different concentrations of saline sodium citrate. Then you dry the slides, apply the counter stain, which is tapping. Okay, tapping, put a cover slip and view with appropriate filters. Okay, so FISH follows the similar steps. So all types of FISH, whether it be from the AF FISH or prenatal testing, whether it is done for prenatal testing or hematological malignancies or your tissue FISH. So mostly they will follow 80% of the steps will be similar, except for some pre-processing that is involved for AF and tissue FISH. Okay. Next. So what are the broad applications? So identification of specific, again, the term specific is important, specific chromosomal abnormalities and interface FISH for abnormalities given failed cytogenetics, sometimes in cultures are very difficult to maintain. Diseases like the myeloma, multiple myeloma, or ALL. So FISH becomes advantageous in those cases. Then monitoring disease progression from time to time, FISH is recommended. Characterization of morphic chromosomes, where the chromosomal origin is not known. So we can go for techniques in FISH with chromosome paints. Okay. Then again monitoring the success of bone marrow transplantation in case of opposite sex donors. Okay. So FISH is used there with XY probes. Okay. in research, identification of new non-random abnormalities, you can check multiple chromosomal rearrangements by using techniques like the multicolor FISH. Okay, and identification of regions of amplification deletion and identification of translocation breakpoints by using techniques like the multicolor mapping. In cancer genetics, so it's used in solid and soft tissue tumors, diagnosis of blood cancers, hematological malignancies, monitoring success of bone marrow transplantation. Coming to tissue FISH, so it has some. So tissue FISH has some additional retreatment steps because you're doing it on FFPE slides, formalin fixed, paraffin embedded slides. Okay. So for tissue FISH, we require the FFPE slide. So you have to do some pretreatment where you remove the paraffin with xylene and ethanol. Okay, followed by heat and enzymatic pretreatment. Okay, heat treatment at a higher temperature in buffers like we have these citric buffers, citric acid buffers, okay, sodium thiocyanate. So all these chemicals can be used in this heat pretreatment. Followed by enzymatic pretreatment. Again, you have to do trypsin or pepsin. Okay, or proteinase. So that depends on the labs methodologies. Followed by after this, the probe addition is that once the DNA is exposed, the probe is added, and all other washing steps are similar. Okay. TAP is again the counter stain. It gives a blue background so that your signals orange, green are clearly visible in contrast. Okay. So you have FFP treatment, you have a xylene treatment, you have auxiliary alcohol treatment. Then you have chemical and protease dilution followed by ethanol dehydration and FISH. So this is the workflow for a typical FFP tissue FISH slide. Okay. So with the meta systems, we have a kit. In-house protocols normally take 4 to 6 hours. Our protocol just take 2 hours with our kit, and you can go up to 50 slides per kit. So it's all very cost effective with our kits. So sample types used in FISH, you have the FFP slides for tissue. Then any other fixed cell suspensions can be used, blood, bone marrow, blood smears. You can do FISH on almost any type of sample. Only thing is a bit of processing is required, and the cells need to be in fixated. Be fixed in the fixated, farmers fixated. And special cells like air, buffalo cells, everything can be fished out. So coming to types of FISH probes, we have the locus specific probes. So because FISH is more specific, so we target a particular gene locus for a disease. Okay. And you have the locus specific probes. We have the translocation. Under this locus specific comes the translocation probe, break apart probe, amplification deletion, centromere enumeration, just for checking the numbers of copy numbers of chromosomes. So that's not centromere enumeration probes. Then you have the chromosome paints, where you can paint the entire chromosome with either green, orange, or blue fluorophores. Then you have the multicolor FISH and multicolor banding probes. Okay, so these two, the first two are much in use. So translocation FISH probes you use when you know the translocation partner. Suppose you know 9 and 22, so you have the BCR ABL probe for that. Suppose you don't know the partner, you just know that one region is getting translocated more often, you go for the break apart probe. For example, in case of MLM, so you have a 11q probe, so break apart probe, which targets the 11q region. Both the fluorophores target a specific region. Okay, so in order to check whether there is a translocation that has happened, so you just know that you get distinct signals. Okay, green or orange signals. Okay. If the signals are fused, it is normal in case of break apart probes. If the signals are distinct, it is abnormal. Okay, the reverse is true for translocation. Translocation, if there's a fusion, the sample is abnormal. If you find distinct signals, it is normal. Okay. Other things are pretty straightforward. Amplification, you will find a lot of green or orange signals. So that is amplification. Deletion, you will find loss of green or orange signals. Next. So this is an example for the VCR ABL1. Okay. So VCR on chromosome 22, ABL1 on chromosome 9. So we have given the normal pattern will be 2 green and 2 orange. Whereas the abnormal, you will get 2 fusions. So you can see fusions here. So 1 green and 1 orange will fuse. It will give a yellowish tinge, which can be observed under a microscope. Okay. So in this case, fusion represents the abnormal pattern. Distinct signals represent normal sample pattern. Next. So coming to break apart, you have the probe designed in such a way that both the fluorophores are present in a particular genetic locus. Okay, so in the same chromosome. So that was the example I was giving about 11q. So similarly, we have other break apart probes. So one fluorophore on one side of the locus, other fluorophore on the other side. So if there's a translocation happening, you will get distinct signals. Okay, so distinct orange and green. So if there's no translocation, you will find fusion signals like this, which represent the normal signal. Okay. So for abnormal, you will find 1 fusion, 1 green and 1 orange. Coming to image. So next is the amplification rule. So as the name suggests, there is going to be an amplification of gene copies. So here, this is an example for HER2 new probe. Okay, so you can see a lot of orange signals here, like a smear. Okay, so those are known as HSRs, homogeneously staining regions. Then you have the control probe, which is in the centromere. It is known as CEP, Centromere Inversion probe. So this HER2 gene is in chromosome 17. So you have something Q12 region targeted here, and the control is the centromere region. So control is green. So if you find 2 green, 2 orange, then it is normal. Okay, sample is normal. If you find group of orange with normal green, then there has been an amplification. Okay, so there are different types of amplifications. You will find DMs. These are known as DMs. This is known as HSRs, homogeneous staining regions. So next. And I will come to how the analysis is done in F2 in the subsequent slides. Coming to deletion, you will find that this is a LU TP53. So 2 genes have been targeted, LU and TP53. And normal cell will have 2 orange, 2 green. And aberrant, you will find that if there is a deletion of TP53, then there will be 1 green, and if there is a deletion of LU, there will be 1 orange. So based on the genetic regions targeted by the color of the fluorophores, you can make the analysis. This is another type of probe known as chromosome paints. So you can see the entire chromosome is painted green. So this is XCP3, chromosome paint 3. So coming to molecular FISH, this involves combinatorial labeling strategy, because we have got 5 fluorophores apart from happy. So you can have 2 power n minus 1 combinations, okay, which is more than around 31 combinations, more to cover the 24 chromosomes. Okay. So next. So this is how the image will metaphase spread will look in a multicolor fish targeted sample. Okay. And that can be used in detection of marker chromosomes. Marker chromosomes, like I was telling you earlier, we don't know the origin of the chromosomes, so we just use paints or multicolor fish technology to know the origin where the chromosome has come. Okay. And detection of translocation. So this the most common use for multicolor fish. So you can detect multiple translocation partners or various chromosomal rearrangements using this technique for multicolor fish, because each chromosome set has a distinct color. Okay, the color is given by the pseudo color is given by the software. So you require the probe kits because there are only 5 chromosomes available. So the extrapolation is done by the software. Extrapolation of 5 into 24. Okay, so you need the software for that and the probe kit. Okay, and this is the whole genome analysis technology. Okay, like your karyotyping. The karyotyping, one thing is it has a higher resolution of 10 MB. Okay, this can go down to 2.5 MB resolution. That is the advantage for using multicolor fish and multicolor branding. So this is just an example. So we can see the 1,9 translocation being detected in one of the blood cancer cases. This is 3B lymphocytic leukemia. Okay, next. And this is our proprietary technology, and multicolor branding, where we have patents, US and European patents for it. Okay. So here you can see that all the bands in a particular chromosome can have a separate or unique color. Okay. So all the 24 colors in a chromosome, single chromosome. So this is mostly for intrachromosomal operations, okay, like inversions or duplications. So all that can be detected to a resolution of 2 to 2.5 MB. So this is more about intrachromosome. So coming to blood cancers or hemato-oncology, we have fish used for fish covers all the disease entities in myeloid and lymphoid cancers. Okay. So AML, ALL, so all the panels. So you can find most of the panels are available with fish for detection, okay, for diagnosis and also for prognosis, okay, and for targeted treatment. Next. So these are the different target regions and their genes that are targeted by fish. In case of MDS, CML, AML, next, CLL, ALL, so various. So it is more specific and it is very easy. It's a relatively easier technology compared to whatever we have been discussing on. Okay, so it's more specific. It's easy for the clinicians to base their diagnosis upon. Okay, so that is an advantage, and it has a very short turnaround time. Next. Next. So here, fish in solid tumors, at least in HER2, it's really gold standard. Okay, for HER2 in primary breast cancers. So approximately 20 to 30% of breast cancer tumors overexpress HER2. Okay, HER2 gene. Okay. So in these cases, based on fish, the treatment modality can be decided. So they provide here known as trastuzumab. Okay, it is a monoclonal antibody for treatment in such cases. Next. And HER2 testing is pretty different from other testing. Most of the amplification probes, so you need to check both the ratio of HER2 and the control region. Okay, so you have the HER2 versus the CEP17. CEP17 is Centromere enumeration probe 17. So this orange to green ratio is to be taken because there can be a pseudo polysomy of the centromere region. Okay, so they normally do the take the ratio in such cases and provide the treatment strategies. Okay. So you can see greater than 2 and less than 2. Less than 2 is normal, greater than 2 is positive or 2 positive. Okay. Next. And for soft tissues, we have different probes. Okay, DWSR1 for any sarcoma and synovial sarcoma. So all these are for soft tissues. That is anti-tissue muscles neurosis. And solid tumors also, we have there are probes, FISH probes targeting various genes. So these are genes in bladder and breast cancer. We have colorectal genes, for eye cancers, head neck cancers, liver, lung. But only a few of those will be you will come across in diagnostic labs, which are mostly cited for the clinicians. Okay? For example, in lung, they have the EGFR or the ROS, ALK, which are targeted by FISH. Okay? And coming to the other role, that is in bone marrow transplantation studies. So we use the XY probe in such cases, and it is used for chimerism studies in opposite sex bone marrow transplant patients. So here the donor and recipient should be of opposite sex. Then only this method can be performed. Which test can be performed using the XY probe. Okay. So if there is the donor is XX, recipient is XY. So you just check whether the XY has relapsed. Okay. Donor is XX. He has given the cells to the bone marrow to the XY. Okay. So if XY cells are more, then the cancer has relapsed. If XX is more, is normal. So that is the study in bone marrow transplant. To check the chimerism. Next. So this is the prenatal. Madam has already discussed all this amniocentesis. We have the chorionic villus sampling and products of conception. Okay, so FISH is done in all these samples. Okay, AF mostly is done, and CVS also. AF is the predominantly used sample choice. Okay, so here also the testing is mostly done for 13, 18, 21 syndromes. Patau's, Edwards, and your Down syndrome or the XY syndromes, Turner's, Klein filters. Okay. Next. So here we have the 13, 18, 21 XY probe. So we have two kits. We have the 13, 18, 21. 13 in green, 18 in blue, 21 in orange. Okay. That is the normally used fluorophore. And we have the XY, X is again green, Y in orange. Okay, so this we'll be doing in the lab today. We will take some samples and we will probe the 13, 18, 21 XY probe and we will check for the cells tomorrow. Okay. So this is mostly for, as I was telling you, to check symptoms. And FISH has also been used in pre-implantation genetic diagnosis, where you remove the embryos at 6 to 8 cell stage. From that, you take one blastomere, do the FISH, okay, check for any abnormalities, then again put the embryo back. Okay, so this is pre-implantation genetic diagnosis. FISH can also be done here, but it requires very skilled embryologist. So there's the help of a skilled embryologist in order to do the fishing, okay, because it's going to be done on blastomeres. Next. So with mechanisms, we have the complete solutions for FISH. We have the one-stop solution, and we have this is our automated scanning system. Okay, so we also have the manual microscopes and we have the softwares for karyotyping and FISH and ICs, which we'll be showing the lab tomorrow. So this, and we have the camera, which is known as full cube, okay, so for capturing images. We partner with Zeiss. Zeiss provides the microscopes and we provide the software. And this is the metaphor or the automated slide scanning and analysis platform. And we have different FISH probe panels to support various cancer types and also prenatal testing. And we have FISH advertiser with us to for easy fishing. Yes. So everything, we have the one-stop solution for FISH. Next. I'll just leave you with a video of our automated scanning and imaging platform. So this is the slide feeder. This cartridge can have up to 80 slides. So up to 800 slides per day can be scanned. Okay, and it takes around 2 minutes for scanning a slide. Okay, 2 or less than 2 minutes for scanning a slide for metapages. So with this, I would like to conclude my talk. Are there any questions?
Any questions? If there's no any questions, we can close. I request everyone to just join for a photo session. We will have a couple of photo sessions here, and then we'll move to our hospital building where we will have lunch and then followed by the workshops. So all the workshops today afternoons and tomorrow's will be conducted in the hospital building. Okay, we'll guide you to go there. After that, we just finish up our photo session. You're going on. We'll just have one photo here and then there in the front, the center hall and.
Will not photo,